Well, hello everybody, and welcome, or welcome back if you were able to join us yesterday. This is the second day of the 2022 Honors Research Conference at Millsaps. My name is Alex Rice. I am a math professor here at Millsaps, and also just as of this past fall, I'm the director of the college's honors program. And in case you weren't able to join us either in person or on YouTube yesterday, I'll just repeat myself a tiny bit and say that this is one of the most valuable and mature academic experiences that we offer here at Millsaps. For the students that are speaking today, this is the culmination of a journey that started in, with a research proposal from the fall of 2020. So they began work on this in fall of 2020, worked all through spring and summer and the fall of 2021. So this is almost a year and a half in the making. A lot of the work they're going to talk about is highly specialized and technical. Some of it is very creative and personal, and they're going to take that year and a half of highly specialized, mature scholarship and try to distill it today into 15 minutes for a general audience. And certainly, as I can, as us as faculty can say from our own experience, that is an exceptionally challenging thing to do. And I am consistently impressed with our undergraduate students' ability to do that. And so I'm looking forward. We have 3 sessions today. They'll each have 3 talks. And hopefully, if all things go well, this is the last you will see of me. The sessions will be moderated by our students. And so I'll introduce our first moderator. She'll be speaking later today, doing in honors in English. But for now, she'll moderate the first session, and that's Brailey Vine.
Hi, welcome. I will be moderating the first session, as Dr. Rice said, and I have the privilege of introducing 3 really great friends of mine. So for the people that are presenting in my session, I just wanted you to know I'm gonna have like a timer going, and when it reaches 15 minutes, that's how long you're supposed to present for, I'm just gonna raise my hand, so if you'll glance over at me every now and then, I'll have my hand raised when you're running out of time, and then we'll have, I think, 5 minutes for questions. So, okay. So without further ado, I'm pleased to pronounce JAILYN NEWELL, our first honors project of the morning, and her project is entitled Hidden in Plain Sight, the Invisibility and Hypervisibility of African American Women in Society.
Good morning. Good morning. My name is JAILYN NEWELL, and I am getting my honors in sociology. The name of my thesis is Hidden in Plain Sight, the Invisibility and Hypervisibility of African American Women in Society. So what this does is it explores the relationship of hypervisibility and invisibility of African American women, and how it contributes to artistic expression, specifically literature produced by African American women. So the key focuses of this research involves the conceptualization of stereotypes and controlling images and collective trauma. Then it explores the relationship between controlling images and collective trauma, specifically arguing if controlling images do render African American women invisible and or hypervisible. It also explores the relationship between the hypervisibility of African American women and artistic expression. My literature review includes the conceptualization of controlling images and the many controlling images that are associated with Black women, both typical and contemporary controlling images. It also includes the conceptualization of collective trauma, specifically the invisibility and hypervisibility of African American women. My methods include a content analysis where I used existing data that probed mainstream media to identify and study themes such as controlling images within mass media to draw inferences about an underlying culture. I also conducted a literary analysis where I selected an author of choice, specifically Jesmyn Ward, and conducted a literary analysis of 2 of her works, Salvage the Bones and Sing Unburied Scene. In the literary analyses, I looked for 3 things: the presence of controlling images, the interplay between invisibility and hypervisibility, and controlling images and forms of resistance, such as emotion management. So controlling images. I conceptualize controlling images as tools of oppression that are used by agents, such as elite white men, white women, and even members of the African American community. I also talked about how controlling images are a socially created narrative of Black womanhood and Black motherhood in some cases. So one of the controlling images that I talked about was the Mamie character, which is an archetype that whites expect all Black women to follow because she is loving and nurturing to her white family. She was also expected to transfer this subordinate behavior to her children. Her sexuality was repressed because she was a heavy-bodied woman, making her asexual. Next is the matriarch. The matriarch was a representation of a failed Mamie because due to her unfemininity, meaning she had some connections to masculinity, which included her being the breadwinner, the head of her household, and also the emasculator of men. The welfare queen. So the welfare queen and/or recipient I talked about is a modern-day example of a breeder based on the overarching idea that welfare mothers had high fertility rates to take advantage of the welfare system. This also caused the sexuality of Black women to become depicted as deviant behavior and become a highly politicized issue. The video vixen. The video vixen was the epitome of the hypersexualization and objectification of the Black female body in urban culture. She used her body for economic profit and social status. Then we also have the fast-tailed girl, which is the hypersexualization and adultification of the Black girl's body and her sexuality. Unlike white girls, Black girls were subjected to demonization when their bodies developed during the pubescent stage. Black girls are also viewed as sexually precocious because they are seen as sexual partners to boys and immediate mothers attributed to allowing men to take advantage of them. So in some form or fashion, the controlling images that I mentioned have connections to masculinity, which does make them appear queer. For example, the Mamie, her asexuality and her body weight. The matriarch, because her overly aggressive nature, she was the emasculator, she was the breadwinner. And the video vixen, where she used her sexuality to her advantage, she sought capital by using body and sexuality as a means of production, and she also equated her body to the empowerment to young Black girls. Now I would talk about the relationship between controlling images and collective trauma, or the framing of invisibility and hypervisibility. So collective trauma is conceptualized as the influence of one's individual trauma upon the community, which in turn thus allows the community to form a collective identity to avoid, heal, and resist from those individual traumas. After the abolishment of slavery, slaves were granted the autonomy to form a collective consciousness, a theory that was formed by Emil Durkheim, where they formed a collective identity or unifying force based on shared experiences from the effects of slavery. This then allowed the group to use forms of code switching as means of survival, another theory used by W.E.B. Du Bois. Black women in this case did do the same thing and engage in code switching as a means of survival to resist controlling images that were produced by gendered racism and other numerous factors. So gendered racism does talk about this social hierarchical order where white elite men are at the top and Black women are at the bottom, or known as the mules of the world. They also experienced a social phenomenon called double jeopardy, where they were not only Black, but they were also women. And they fell at the intersectionality of feminist movement and anti-racist movements. With that being said, controlling images, I argue, did render African American women invisible and hypervisible. Invisibility and hypervisibility are intertwined to understand how white spaces construct the narrative of Black womanhood and motherhood. Hypervisibility is the observation of something that is consistent with being put on display. Invisibility is the reaction of being placed in a subordinate position. Black women are rendered hypervisible because of the policing of their sexuality and behavior to create this central narrative of Black womanhood, while it also renders them invisible because it forces them to engage in emotion management like coping or the suppression of emotions. So my data results and finding. The author of choice I did choose was Jessamine Ward. I chose her because she is from the state of Mississippi and also because her works have not been heavily researched upon, like Maya Angelou and Toni Morrison, and etc. The two works that I focused on were Salvage the Bones and Sing Unburied Sing. In Salvage the Bones, I focused on two characters, Esh and Mama. Esh and Mama did exhibit qualities and characteristics of controlling images that rendered them invisible and hypervisible. Esh was the fast-tailed girl, Mama was the savior. Esh was the only character that engaged in forms of resistance, specifically using her sexual experiences to fill the temporary void of security that she lost when her mom died. In Sing Unburied Sing, I've also focused on two characters, Leonie and Ma'am, who also exhibited behaviors associated with controlling images that rendered them invisible and hypervisible. Leonie was the negligent and irresponsible mother that engaged in drug abuse as a form of emotion management to cope with the loss of her brother and later the loss of her mother. Ma'am was a religious figure that brought in notions of spirituality and religiosity. This connection emphasizes the role that African American women play in their community, how they made a way out of no way. But they were not afforded the same luxury of receiving help from her community during her time of need. Ma'am was also significant because of her knowledge of faith and healing that allowed me to flesh out connections between Black women and religion and health. So in my discussion, Ward does not use her artistic expression to reinforce controlling images, but she uses them to dismantle the social and economic injustices they reproduce. She transformed these images into three-dimensional characters to highlight the contradictions of these controlling images. Taking advantage of being an insider and an outsider, she resists the notions of controlling images to push back against them and redefine how we understand the narrative of Black womanhood. She does utilize controlling images, but she only uses them when referencing emotion management like coping. She shows how Black women are perceived as strong. It's just because they have to be. So in conclusion, I aim to portray how Black women use artistic expression, specifically literature, as social locations to dismantle these distortions of reality and injustices. Although controlling images do render African American women invisible and hypervisible, it does not deprive African American women of having a voice to heal, to resist, and to avoid these stereotypes. It instead gives them the opportunity to be agents of knowledge, to use critical and alternative paradigms to avoid, resist, and heal from controlling images. As best stated by Patricia Hill Collins in Black Feminist Epistemology, the existence of a self-defined Black women's standpoint using Black feminist epistemology calls into question the content of what currently passes truth and simultaneously challenges the process of arriving at that truth. Thank you. Also giving special thanks to Dr. LaWanda Evans, who was my advisor, Dr. Ian McMaster, who was my second reader, Professor Sue Carey Drummond, who was my third reader, and Dr. Alex Rice, who was director of this entire conference. Thank you. Okay, that's cool.
All right, we have some time for questions. Is this on?
Don't be shy.
Can you tell me about the interaction between your own lived experience and the project that you're working on? I know that probably provided inspiration, but it's also potentially a barrier towards objectivity.
So it definitely, it was very personal in so many ways, especially when I was reading Sing Unburied Sing. It made me think about my grandmother. And so Ma'am was this character that brought in notions of spirituality, religiosity, and being a Black woman in her community where she offered help but didn't have the luxury to do the same. So my grandmother is 80 years old, and we're not far removed from the Civil Rights Movement. And while I was reading this book, I was asking her, some really major questions like, did you do this? Did you, make herbal teas and give them to the community? Did you, in any form of fashion, were you this person that I'm reading about? And, she provided as many answers as she could. So, yeah, it hit home in so many ways.
From your research, from your life experiences, can you tell us how what you read, how you can internalize it and bring out the real truth? How do we go forward?
So that's a very good question, and it's also a very hard question to answer because it requires everyone opening their ears, but it also requires everyone opening their minds. Everyone can create a lot of solutions, but no one, takes the time to consider those solutions to implement so many things that, our Black community needs. And it goes from a social issue, it goes from an economic issue, it even comes from a political issue. All these things just play in the role of how African Americans are viewed and how these stereotypes and controlling images are reproduced in mainstream media. So what we know the narrative of African American women to be today is not exactly true because, of course, being an African American, we know what is real and we know what is not. But however, because of the role we play socially, economically, and politically, it forces us to engage in forms of resistance, which is basically, trying to push back against all of these things that may not be so true. And where we suppress a lot of feelings, we suppress emotions, we suppress everything else, and we really don't have an outlet to let those things flesh out, which is why so many women, so many African American women, men, boys, girls are entering into mainstream media using books, music, art, using so many artistic expressions, but other forms as well to talk about these experiences and validate them in their own way.
Are there any more questions? All right, if not, let's give another round of applause for JAILYN. And next, we have our second presentation by Faith Gilmore, and her project is entitled A Slave to Lynching.
Good morning. My name is Faith Gilmore, and I will be doing a presentation in the field of history titled A Slave to Lynching. So I chose to conduct my research in history because I have a passion for African American history as well as the law, and I wanted to be able to incorporate both and create a piece that I could be proud of. And although African American history is full of pain, I feel as though I have a duty to be the voice and tell the story my ancestors were not privy to doing. So history has a tendency to repeat itself through evolution, as we have seen the presentation of racism and racial actions in the U.S., which have transitioned from slavery, the Reconstruction, and Jim Crow era, also to modern-day society. In each era, the racial violent actions of enslavement, disenfranchisement, lynching, and burning of churches, they were viewed differently from a legal standpoint, often not tied to clear consequences for their perpetrators. Although society has changed from a historical perspective, the mentality of racism is still the same but expressed differently. In my thesis, I argue that earlier legislation, which once took proactive measures in implementing laws ensuring white supremacy continued to stand firm in the South, has now become reactive in protecting Black Americans after they received the full scope of freedom due. Legislators' lack of approach of anti-lynching laws since 1918 to 2019 was a prime example of how motivational racism and was given permission to fester. It took a drastic and unrelatable event to occur towards white Americans before the justice system properly recognized lynching as a hate crime and to enforce laws to prevent history from repeating itself. For instance, with early legislation, I wanted to start with the 14th Amendment, which granted citizenship to those born in the U.S. and former slaves, allowing Black bodies to become recognized as Black Americans. However, the amendment did not protect Black Americans in the South because they were continuously being oppressed and dehumanized. So with the 15th Amendment, where we're establishing boundaries to protect voters' rights. Voters were not to be denied the right to vote due to race, color, or prior condition of servitude. Yet, we have the introduction of literacy tests in the South, which were a tool used to silence and prevent Blacks from registering to vote. And those who tried were either killed or they lost their property due to destruction. So we enter to this era of lynching. Lynching is defined as having several people come together to use an extra-legal method to put a person to death without permission of the courts or legal sanction based on their own or traditional justifications. Lynching began after the American Revolution in 1775 and was derived from the brother of the man who founded Lynchburg, Virginia. So after the Revolutionary War, lynching was used as a non-lethal form of embarrassment for white descent. However, as we gravitate towards the Civil War, lynching is transitioned to a more lethal form of punishment where Black Americans were often killed due to their new status of Black Americans successfully fighting for their freedom. Lynching was considered capital punishment in an independent justice system where lynchers and lynch mobs were viewed as heroes, saving their communities from the terror of, at that time, Black Americans or also Black bodies. It was a form of public sovereignty and self-policing and the reign of justice being controlled by citizens. I view lynching as being more than just a rope and tree. It is a mutilation, dismemberment, torture, and dragging of Black Americans regardless of age and gender. The disposal of the bodies was done with no regards for victims as a person, someone with an identity, or just being that of a human being. The passing of anti-lynching bills was once considered being rejected by Southern legislators. Those 200 anti-lynching bills that were proposed to become future laws were to make lynching illegal. However, every bill that passed the House would go to the Senate chambers, and they would often die because they were controlled by the Southern Democrats, formerly known as Dixiecrats. Over the gradual change of each era, the pattern of using systemic methods to control the lives of Black Americans changed because of the laws being enacted at the time, but the philosophy was planted in the next time period, just in a different form. And so that new form is considered hate crimes, which are defined as being violent and nonviolent crimes committed against a select group of people based on their characteristics such as ethnicity, religion, sexual orientation, and other features that set them apart. During earlier times, the hate crimes that we consider today were characterized as a violation of one's civil rights, which were based on their race, color, religion, or origin. Today, we only have 47 states that recognize hate crimes and have their own state hate crime laws. And those states that do not are Wyoming, Arkansas, and South Carolina. So I chose to focus on the murders of James Barrett Jr. and Matthew Shepard, which were groundbreaking events that motioned our legal system to act against hate crimes. So with James Barrett Jr., he was killed June 6th of '98 in Jasper, Texas, a Black American man that was offered a ride by 3 white Americans, and not only was he beaten, but dragged miles down the road, and the remnants of his body was left in front of a predominantly Black church. This was a hate crime that shed light on the issue of racism and modern-day lynching. It opened a historical wound that America has yet to heal from. And with Matthew Shepard succumbing to his injuries on October 6th in '98 in Laramie, Wyoming, he was a white American college student that was robbed, beaten, pistol whipped, and tied to a fence, all because of his sexual orientation. The murder of Matthew Shepard became more personable to the public because they were able to put themselves in that situation where they viewed him as being that a neighbor, a friend, or someone that they love as their son or themselves. And this was a murder that white Americans had never experienced, nor could they relate to. And so we have the introduction of the Matthew Shepard and James Barrett Jr. Act of '09, which is our federal crime law that makes it illegal, not only does it criminalize, but it prosecutes a person who harms or attempts to harm said other based on their federally protected class, which include but are not limited to race, color, religion, national origin, gender orientation, sexual orientation, identity, or disability. And as you can see, it took 11 years for federal hate crime laws to be passed, which is yet another example of how our legislative system exhibits reactive behavior when it comes to protecting its people. And so I wanted to focus more so on Mississippi and our state code 1972. The sections that refer to hate crime laws were revised to include newer classes of victims and create a felony or misdemeanor that were committed to a person's classification based on race, color, ancestry, ethnicity, religion, national origin, gender, or any crime committed towards a first responder based on their duties, which is fairly new. As you can see, lynching was not mentioned as a hate crime in the Mississippi Code. That was because lynching did not become a part of the qualifications for a hate crime until 2019, where our 116th U.S. Congress passed H.R. 35, Emmett Till Anti-Lynching Act, a bill that acknowledged lynching to be the ultimate expression of racism in the United States. Lynching was described by Congress itself as a tool used to interfere with the federally protected rights. If our legislative system continued to take preventative measures like it once did before the right reasons, then maybe someone else's family would not have had to experience a loss because of their identification within a protected class. A lot of lynchings that took place, such as the Emmett Till, the 3 civil rights workers, Matthew Shepard, James Barrett Jr., and a host of other victims would have lived a much longer life, which is a reason why we're left asking, what if? And so, closer to our conclusion, we have the fact that as we exited one era and entered another, we witnessed the introduction of different legislative laws that were placed. However, even though Black Americans were given that inch of freedom, they ensured that there were loopholes within a clause that gave the old mentality of America, which was white supremacy, the permission to continue to exist. We began to see Southern white Americans develop the philosophy of public sovereignty, where they negate the laws and take it upon themselves to self-police their community. This is where lynching has become the ultimate and extreme expression of racism. And with the progression of lynching, it also led to hate crimes being recognized as not only impacting Black Americans, but white Americans as well, and others based on their characteristics, which boldly sets them apart. I personally believe that America is still a slave to lynching. This time, it's not just among the races, Black versus white, but also Black versus blue, where we have officers that not only use force, excessive force and tactics to murder unarmed Black Americans, but there are white Americans that are police officers that are still slaves to the mentality of fearing their Black Americans, especially men, by needing to assert dominance. So, the future of our history. Although we are beginning to witness our national lawmakers make more decisions and incorporating the demands of modern times by the removal of Confederate statues, renaming the street that leads to the White House as Black Lives Matter Plaza, and holding those accountable for their actions that are in authoritative positions, I want you to ask yourself, can we truly say that it is enough? Are we as a country, as Black Americans, taking the measures to prevent the next generation from experiencing this painful history of lynching, or is it too late? I would also like to acknowledge Dr. Rolfe, Dr. Evans, and Dr. Crenshaw for helping me with my project and giving me the free range of being able to construct a piece, a historical piece to me that I can be proud of.
Are there any questions? KAYLAH?
First, great job. In your project, you mentioned how the Black versus blue, how there's, a great tension between the Black community and police officers. But when you mentioned how there's this fear within Black police officers and how they view Black people, could you talk more about that? And maybe are those controlling images and how they perceive Black men or Black women?
So I agree that it kind of is a controlling image, because we have to think about the timeline of once African Americans received that freedom due, that we see that white supremacy still reigned free within the land, but they did public sovereignty and self-community policing. And so once they kind of got that sense of becoming police officers, which now they can actually do it legally and it's protected by a shield, that it still is a problem.
Did you run across any particular examples of legislation at the state's levels or the federal level that you really felt does help move the needle in terms of systemic?
I would would say that the neural one with George Floyd, which happened in 2020 but was passed in 21, I would say that's a new step in a new direction because we're not waiting 11 years for a new form of a bill to help Black Americans. So I would say the George Floyd bill and that was passed in 21 actually is helping a lot more.
If there are no further questions, let's give her another round of applause. And for our last project of the session, we have Jeremy JUVE presenting his honors project entitled Investigating the Role of Fat Metabolism in Germline Tumor Development in C. elegans.
Hello everyone, my name is Jeremy JUVE, and like Brailey said, my project is on investigating the role of fat metabolism in germline tumor development in C. elegans. That's a lot. We'll get through it, I promise. So why does any of that matter? According to the National Cancer Institute, 40% of Americans will be diagnosed with cancer at some point in their life. And there's over 600,000 cancer deaths in the US annually. So anything that furthers our understanding of oncogenesis, tumor development, etc., can help lead to new treatments, can help reduce the death rate, and can, in general, hopefully, eventually, lead to a universal cure for cancer. Not saying that my project does, but it helps a little. So, why fat metabolism? There are several known links between cancer and fat metabolism. Most of you probably know that obesity leads to a higher risk of developing cancer. And also, fat metabolism provides all of the large amounts of energy that are needed for cancer cells to continue proliferating and develop a large tumor. Sorry, I have asthma. So cancer could also affect metabolism more directly. There's a specific example known as the Warburg effect, where cell metabolism is actually altered to not use oxygen even when it's available. And as to this project in particular, what initially inspired it was some prior work by one of the postdocs in the lab that I worked in, which involved depleting FAT6, which is a gene that's required to build up a fat storage. Like if you don't have it, you just will not have any body fat. And he noticed when he depleted it that there was no fat storage, and also the tumor sizes had shrunk massively. So what that inspired this project, we are now trying to find other methods of disrupting fat metabolism that are less harmful, because you can't just prevent people from having any body fat. You do need some of it to live. So we are trying to find other targets besides FAT6 that we can use. And how are we doing that? According to the central dogma of molecular biology, everything in the body is ultimately the result of DNA being transcribed into messenger RNA or mRNA, which is then translated and assembled into a protein which performs most of the functions required for life in the cell. We can actually disrupt this process quite easily, right there. So there's existing mechanisms to regulate gene expression in all living organisms, and we can take advantage of one known as post-transcriptional gene silencing, which is when cells that have already been transcribed from DNA to mRNA can still be deactivated. And we're going to get more into the specifics later on, I promise, but for now, just understand that we can use this to stop the protein from being built. So, a protein does something, you take out the protein, that doesn't get done. I know we're not biologists, well, most of us anyways, but that's the basic explanation. And so we do this in nematodes. So C. elegans is a small nematode, it's about 1 millimeter long, and it just lives in soil. Go out there, grab a handful of dirt, there's probably nematodes in it. And C. elegans is actually really special. It's a model organism. It's used all over the world for all kinds of research. It's how we learned how cells die. It's how we learned how cancer first starts. You'd be amazed at what C. elegans has taught us. And the reason it's so useful is because 83% of its genes are in humans. With a similar function, similar structure, it's perfect for doing research. And for this project in particular, we get two very beneficial aspects from using C. elegans. The first one is that C. elegans eats bacteria, which enables us to do a protocol called feeding-based RNA interference. And I have a whole slide dedicated to explaining what those words mean later on. And then it also has a large germline. There's a slight issue with doing cancer research in C. elegans. The only cells that continue to divide once the nematodes reach adulthood is the germline, like the sperm and the eggs. So the only place that you can induce cancer in nematodes is in the germline. But C. elegans in particular has a very large one. So if you induce a tumor in it, it's very large and you can just see it under a microscope. And here's just a little animation of a C. elegans crawling around under a microscope. They're just a tiny little worm. So how do we give nematodes cancer? So nematodes are diploid. That means, like us, they have two copies of all of their DNA. So, given Mendelian genetics, you inherit half from each parent. You have like a quarter chance of every possible combination. So when you mix a GOLD1 knockout mutation, GOLD1 is a gene that regulates meiosis. So cells go from mitotic division, where they're just dividing, to meiosis, where will eventually differentiate into embryos or sperm. C. elegans is actually a hermaphrodite. It makes both, and it actually impregnates itself with both. But using this GOLD1 meiotic regulator, we can use it to do a double knockout, which is when both of the genes have been mutated to not work, and that 25% who inherit that combination, so only 25% of them actually have cancer. Should buy my inhaler. So 25% of them will get cancer. And the way we tell which one has cancer is we include another gene right next to it called UNC32, which makes them unable to coordinate movement, so they'll just wiggle around in place no matter what you do to them. And I assure you, if you start tapping a plate of nematodes on the table, they'll all start moving around and you'll immediately be able to see which ones are cancerous. So if you look at the photo on the right, the top image is just a C. elegans and its two gonads. And then the second photo is a close-up of a normal C. elegans gonad. You can see the mitotic division in blue transitions to meiosis, which then transitions into sperm and oocytes that fertilize and make an embryo. In a GOLD1 knockout, they instead go from mitosis, briefly enter meiosis, then revert back to mitosis and continue just dividing forever until the host nematode dies. And that gives us a nematode with cancer. And an interesting side effect of that brief entry into meiosis is that it actually develops a teratoma, which is just a region of trans-differentiated cells, meaning just cells that shouldn't be where they are. So you'll get muscle cells or neurons or just whatever cell type shouldn't be there will be there. So how does fat metabolism work in C. elegans? There were 400 genes. Nobody has time for that. So what we do is we take a transcription factor. Transcription factors are special genes that regulate the expression of other genes. So for example, you might have one transcription factor regulates 20 others. So by influencing that transcription factor, you can influence the expression of 20 other genes with the time and effort of one. So in C. elegans, most of the transcription factors related to fat metabolism are known. So I performed my experiments on these 5 in particular. You don't really need to worry about the names. I'll explain which ones are the most important later. But just know that all 5 of them can be found in human. They all have a human equivalent of some kind, even if it's not the same name. And I was going to do more, but sadly this was only a 10-week program where I actually had a lab bench to work at. And so I only had time to do these 5. So what I did with these genes is I knocked down their expression in the cancerous nematodes, and then I measured their tumor size. And I also used the FAT6 gene, which if you recall was the gene that initially inspired this project, as a positive control, because every single time you successfully knock it down, you will visibly be able to tell. So it's an excellent positive control to verify that your experiment worked. So how do we disrupt fat metabolism? A couple slides ago, I mentioned that I use RNA interference because it's very easy to do in C. elegans. So RNA interference is a mechanism that allows us to utilize the existing gene regulatory pathways to influence gene expression. We just do some basic DNA cloning by taking E. coli with the gene that we want to knock down and a complementary sequence. We induce it, and it will be transcribed into double-stranded RNA, which we then just put the worms on the bacteria, they just eat it. Once they've had their fill, they will convert the double-stranded RNA into small interfering RNA, and then using some proteins that already exist in most living organisms, they will bind to the target mRNA. For our gene, it will degrade, no protein. So the cell has now made mRNA, and we have now destroyed it. So for every single time we transcribe our target gene with RNAi, that's one instance where it will not become a protein. And we also include a negative control where we just didn't do RNAi on a gene to compare as our baseline. So now, to measure tumor size, we use something called DAPI staining, which is a fluorescent stain that binds to DNA. By staining the DNA, we can see the size of the tumor because the tumorous region will have extra DNA from all of the tumorous cells just dividing constantly. So we use a tool called ImageJ, and we can literally just measure the area of the tumor. So on the left is a totally normal, no cancer, just wild type gonad in C. elegans. On the bottom, we have the mitotic region, which becomes the meiotic region, and then it loops up and around to become embryos. On the right, we have a cancerous GOLD1 mutant, where we have the tumorous gonad, and I literally just circle, like you see here in red, and measure the area. It's orientation specific. We need to take photos of the ones specifically on their side, completely visible. It's not overlapping with itself. But most of them, you could get at least one good photo. And here on the right is just the reference to the earlier slide where you can see the wild type versus the cancerous mutant. And so here are some images of DAPI staining. So you can see in the top left is just our control. The top right is our FAT6. It's kind of hard to tell on like these slides because the orientation just isn't the same. But I promise you, I do have statistical data to prove that these are actually different sizes. And then on the bottom, we have 2 of our experimental genes. These happen to be the ones that are most effective, which you can see here. So we have our FAT6 positive control, but then 3 of our 5 genes that I did this work on significantly reduce tumor size when you knock down their expression. And these are NHR49, NHR80, and DAF2. 2 of the genes didn't do anything. And for reference, the asterisk indicates statistical significance, and S indicates no significance. The solid bar at 1 is our baseline. So the further away from that, the larger the difference in tumor size. So we are massively disrupting fat metabolism because not only are we disrupting a single gene, but that gene being a transcription factor controls however many other genes. So because of this, we want to see what's happening to the fat content of the nematodes. And we're specifically worried about the worm itself, not the tumor. We don't care about the fat content of the tumor. So we use another stain called oil red O, or ORO, which stains triglycerides and lipids. And again, we use image J to quantify this, but instead of measuring area, we measure light intensity. So if you look at the nematode over on the right, we circle this area in image J because it's the entirety of the worm that is not tumorous. There will be no interference from the cancerous tumor, and it's just a good baseline reading of fat content in the worm. And what we do is we just invert the color and measure the intensity of that area minus the intensity of the background, and that gives us a rough estimate of the fat content relative to the baseline. So here's some examples of fat content. If you look in the top right, you can very clearly see what I mean about FAT6. You can just immediately look at that and tell that it's FAT6 because it looks nothing like any of the other ones because it just almost doesn't have any fat in it. Versus on the left, which is our baseline, and then in the bottom 2, you can see that there's no obvious differences in fat content, but I'm afraid the actual data on that is a little more complicated. So, in short, 3 of our genes were statistically significant, with NHR80 actually giving us a 27% reduction in tumor size, followed closely by NHR49 at 19%. DAF2 is at 7%, and then DAF12 again, not significant, but fat data is complicated. So I only had 10 weeks in the lab to actually run this experiment, and there just was not enough time to collect enough data to do statistical analysis on the fat content, except on DAF12 and DAF16, which were insignificant both for tumor size and fat. But on NHR49, NHR80, and DAF2, I do not have statistical analysis on the data because enough data simply isn't there. But for what we do have, now, big grain of salt, like I said, not enough data to be sure of this. But we have 0% change in NHR49 and only a 6% reduction in NHR80. So we get a 27% reduction in tumor size for a 6% reduction in fat content. So what do we do with this? Well, if you recall, I said NHR49 and NHR80 are our 2 best candidates. Those 2 are nuclear hormone receptors, which are a special type of gene that are on the nucleus, and they accept signals from the They accept hormones, from the endocrine system, which can control all kinds of cell functions, Like, if it exists, a nuclear hormone receptor can probably influence it in some way. So these have a wide range of function. And we want to know why specifically do our 2 nuclear hormone receptors reduce tumor size? And then we want to figure out what specific genes reduce tumor size. Because if you recall, transcription factors can influence a number of genes. It's not necessarily a transcription factor itself, it's one or some combination of the genes that they regulate. We also need to go back and redo the fat content data to get enough to draw statistically significant conclusions. And ideally, we need to study more transcription factors. So I'd like to thank everyone in Lee Lab, including Amresh, Sylvia, and my benchmate Iris. I'd like to thank Dr. Rice for organizing this, as well as Dr. Hussa for being my advisor, Dr. Engel and Dr. Toyota for sitting on my committee. And then finally, I'd like to thank Wormbase for providing just countless bits of useful information on C. elegans, the C. elegans Genetics Center for providing the nematodes that were used, and finally, the Cornell MBG program, which enabled me to do the entirety of this project, as well as the NSF, which funded said program.
Does anyone smart enough to understand all that have a question?
Or an asthma inhaler.
I am not smart enough to understand all that. I'm going to ask the question anyway. First of all, I think you did a fantastic job of bringing this down to someone with very little biology experience can understand. So thank you. I had trouble recognizing how you recognize the tumors from someone who's not used to seeing those images. I could not have drawn those little red boundaries around the tumor. Is it fluorescence intensity, density of the fluorophores? What is it that lets you know that there's a tumor?
Yeah, I can actually show you. So you see on, where is it? So you see on the top how you have the 2 gonads and they kind of look like elongated U's? Well, if you go back to the DAPI photos, you can see again you have your 2 elongated U's, which are way brighter than everything around them, and that's because the sheer amount of DNA from all of the tumorous cells makes it stain way more visibly than all of the non-tumorous cells around it. So that whole white area is tumor.
I have a question. When it comes down to your fat cell line, you said knocking out fat 6 got rid of basically all the fat in the body. So are you able to control which fat line you're going after? Are you going after visceral, parietal, white fat, or yellow fat?
No, we do not have specific control over it, and in future experiments, once we've identified a specific gene, there will be more thorough and specific assays to determine the lasting effects on fat content. But since this was a preliminary screening, we just wanted to see, like, overall, in general, is this disruptive enough to be potentially lethal? Well, ideally, we wouldn't disrupt fat content. We would disrupt whatever allows the tumors to utilize that fat as energy. So ideally, like, it would have no other effect on the body except the cancer would not be able to use your body's fat stores for energy. Anyone else?
I wanted to ask about, I guess, the institutions that you said were significant, how the hormone receptors, it seemed like there would be like general hormone receptors, so I was just wondering if you, in future studies, were going to look at kind of the unintended side effects of those receptors.
Yes, well, since those are transcription factors, ideally we'll find whatever gene regulated by them, which probably won't be a hormone receptor. It'll just be, you know, whatever, like it receives the hormone and then based on that, it regulates a ton of other things. Ideally, instead of disrupting something as important as a nuclear hormone receptor, it'll just be whatever minor gene downstream of it that actually has the effects on tumor development.
If there are no more questions, let's give them another round of applause. And we're going to take a short break, so we'll resume at 10:45. Please go get more food and drinks, and thank you for coming.
everyone. My name's Hannah O'Brien, and I'm going to be moderating our second session for today. And we have three great speakers who are going to be presenting during this section. And we're going to start with Christopher BRUNI, who has an honors in biochemistry, and the title of his thesis is Synthesis of Allosteric HIV Integrase Inhibitors based on a Pyridine Pharmacophore. So everyone please give a round of applause for Chris.
Good morning. As Hannah said, my name is Christopher BRUNI, and today I have the privilege of presenting to you all my honors thesis, which is the synthesis of allosteric HIV integrase inhibitors based on a pyridine pharmacophore. And so I know that sounds like a mouthful, but hopefully by the end of this presentation, you'll understand exactly what that means. So to start off, I would like to recognize a few undergraduates that are currently working on the project and others that have worked on this project, those being myself, Sarah Hayek, and Hannah Madke, as well as Sharon Suffern and Victor Michu, and Dr. Kramer is the head professor over us all. And this is also a collaborative project with Drs. Kessel, Dr. Pigza, and Dr. Donahue of USM. So a few things about HIV. HIV stands for Human Immunodeficiency Virus. It is one of the world's leading infectious killers. What it does is it basically targets and kills host immune cells. It is spread through exposure to bodily fluids from an infected individual, primarily through unprotected sex or through the sharing of injection equipment. Stage 3 HIV is classified as AIDS, and in this stage, the host immune system has been severely compromised to where they are basically unable to defend off any infection that they might incur. As of 2020, around 37.7 million people worldwide live with HIV, and since the first AIDS case in 1981, around almost 40 million people have died. In 2020 alone, 680,000 people have died worldwide of AIDS, and as of 2019, the CDC estimates around 1.2 million people in the US live with HIV, and there's currently no known cure for AIDS. So what is the point of studying this if people can live with the virus? And the reason being is that these infected individuals have to take medication, but HIV, like other viruses and bacteria, can develop resistance to this medication. In fact, a new strain has been found in the Netherlands this month, which basically decreases time until AIDS is developed. So typically it takes around 8 to 10 years for HIV to develop into AIDS, whereas with this new variant, it only takes 2 to 3 years. So it's a much quicker onset of AIDS. The researchers believe this variant most likely arose from de novo mutations, which a de novo mutation is basically a genetic change that occurs when you produce gametes, so you produce sperm and egg. And thankfully with treatment, there appears to be no difference in the CD4 cell count or the immune cell count or mortality after treatment compared to the other strains of HIV. And so since I'm making an inhibitor, which is a drug, I found it fitting to talk about the drug development process. I'm not going to go into each step. Just know that this is what every pharmaceutical company has to do with every drug that they come out with. The process takes around 10 to 12 years. It can cost upwards of $2 billion. And so the steps that we have done and are currently doing in the Millsaps lab, we, find a problem, our problem being HIV. Next, we would select the target. So the three main targets that you would select from are oligonucleotides, which are DNA, enzymes, which is a protein that basically does a job, and a receptor that basically is a protein that binds a ligand to elicit a response. So our target is the integrase, the HIV enzyme integrase. Next, we would run some assays, and so Dr. Kessel at USM runs our assays, and we are currently working to find a successful drug candidate in our lab. And so another reason I wanted to talk about this was because Moderna actually just came out with a HIV vaccine that is currently in phase 1 clinical trials. And so this vaccine utilizes the same mRNA technology that the COVID vaccine uses. And the first patient received their first dose at the George Washington School of Medicine in Washington, D.C. as of January 27th last month. And this is not the first HIV vaccine to be developed. All the other HIV vaccines that were developed have failed in clinical trials, but hopefully this one will make it through. So these graphs here basically indicate the prevalence of HIV in our home country as well as our home state. The darker colors indicate a higher rate of HIV diagnosis, so you can see HIV is more prevalent along the southern states, and in our home state is more prevalent along the Delta region. And according to this chart, in 2017 there were more than 381 new HIV diagnoses in Hinds County. So this is the structure of HIV. It looks like a lot, and I just want to focus on a few things here. I would like to first focus on the three main viral enzymes that the virus requires to replicate itself, those being reverse transcriptase, integrase, and protease. I also want to point out this viral RNA genome, and what this genome is it basically contains all the information needed to make a new viral particle. I also want to point out this dark blue viral capsid. This is basically what gets injected into the immune cell to start the infection. as you can see in this next slide. So how does HIV enter an immune cell? It basically binds to the immune cell, injects that viral capsid I just showed you, and once that capsid is injected in, the reverse transcriptase enzyme reverse transcribes that viral mRNA into DNA. That viral DNA then flows to the host nucleus, where the integrase enzyme of HIV integrates that viral DNA with the host DNA. So then when the immune cell undergoes transcription and translation like every cell does in your body, it's basically transcribing and translating that viral DNA into viral proteins, which are then assembled, packaged, and then released, as you can see here, as a new infectious viral particle to go on and infect another immune cell. So this project is working to inhibit the integration of the viral genome with the host genome. And so a few things about the integrase enzyme. It's a tetramer, which means that it's made up of 4 subunits. So these sites outlined here by the red circles are the active site of the enzyme. And what an active site is, it's basically the region of an enzyme where a substrate binds to undergo a chemical reaction. And so a competitive inhibitor is a molecule that binds to these sites and basically blocks it, so the substrate cannot bind to the active site and therefore no reaction can occur. But the active site can also be closed by basically changing the 3D structure of the enzyme by binding elsewhere. And those sites that it can bind elsewhere are known as allosteric sites. So we are synthesizing an allosteric integrase inhibitor, which is also called an ALINI. That stands for allosteric integrase inhibitor, because we are binding to the allosteric sites and we are working to inhibit the integrase enzyme of HIV. So how do we know what to model our drug after? It's important to know the amino acid residues of the allosteric site, as you want your drug to have very favorable intermolecular interactions with these amino acid residues, so the inhibitor will remain bound and thus maintain inhibiting the enzyme. So like I said before, this is a collaborative project. As you can see, these molecules, the other professors we are synthesizing these molecules to basically give to Dr. Kessel to run in an assay. And so the pharmacophores that we are using are the quinoline and pyridine. And we at the Millsaps lab here are trying to synthesize this pyridine pharmacophore outlined in black. So a pharmacophore is the part of a drug that is required for activity. And we can modify it to possibly increase inhibition by substituting substituents into these R groups. And the main differences between our pharmacophore that we are trying to make here at Millsaps and the other ones is the quinoline has this additional 6-member ring, as well as other spots that you can substitute other groups on to possibly increase inhibition. So this is an overview of our synthesis. As you can see, it's a multi-step organic synthesis. It consists roughly of 9 steps. So an organic synthesis is like Lego, but it has a few more rules. And so we are starting for something small, like some Lego bricks, which would be our beginning right here. And we are basically adding on to it to build something more complicated, a more complex molecule, as in this case, the Death Star. And so the molecule that our Death Star that we are trying to synthesize is shown here in black is this pharmacophore. And so some specific aims for our pathway is, 1, to identify reproducible procedures for the initial steps. So those are shown here, the first 3 steps. The first 2 steps are to promote the cyclization of this pyridine ring shown here, and the third step is to chlorinate the 4 and 6 positions on that ring. And we need to optimize those steps and characterize the product. So we need to optimize the yield for those first few steps, because we need a lot of starting material to continue to carry on the synthesis in the later steps. And what I mean by characterize the products is basically to determine the structure using NMR spectroscopy. And so third, we need to explore and perform options for the side chain elongation. So the pharmacophore is outlined in blue here. This is the side chain that basically steps 4 through 8 are working on. We need to, as you can see in each step, the side chain is changing ever so slightly. We need to basically elongate the side chain by carbon, as well as make sure it is in the correct orientation and oxidative state to ensure that it has proper intermolecular interactions and thus the best inhibition. So instead of boring you with going into detail with each step, I would like to let you step into my shoes to perform the third step, the chlorination step, and show you just how complicated it can be. And I know that I just compared an organic synthesis to Lego, but in reality, it's not like Lego where you just click pieces together. There's a whole lot more that it actually entails. And so, like I said before, this is the chlorination step. So we will be using phosphorus oxychloride to replace these hydroxy groups with chlorines. And so this is what your lab journal will look like. You would date it to write the entry number. I've got done about 116 separate experiments now. You write out the reaction scheme, and then you would basically put a procedure. And so the procedure being 14 equivalents, we're going to add 14 molar equivalents of POCl3, heat it to 100 degrees Celsius for 3 hours. Afterward, we're going to quench it with ice, adjust the pH to 7 using sodium hydroxide. Then after that, extract it using methylene chloride, dry it, rotavap it, and what we should collect is yellow oil. So this procedure can basically be broken down into 4 steps listed here on the bottom. So first is the reaction. So after you set up your lab journal, you're going to put on gloves and goggles because safety is the top priority in the labs here at Millsaps. And then you will need to set up the reaction apparatus in the fume hood, as these reactions can give off toxic fumes. So the apparatus consists of an oil bath on a hot plate with a round bottom flask in that is attached to a ring stand, as you can see here. After that, you would then need to calculate the molar equivalents of the reactants you would use, in this case, this molecule here, as well as the POCO3. And once you do that, you would add both to the round bottom flask, and then you would heat it to the desired temp, 100 degrees Celsius for 3 hours for this step. And so what we are doing in this is basically sealing off the reaction to the air, as POCO3 will react with the water vapor in the air, and we are heating it to give it the energy to react. Next, we would inactivate the POCO3, so we add POCO3 in excess. And so we will take the contents of that round bottom flask, put it into a 1 liter beaker, and add 600 milliliters of ice water. And so you want to be sure to stir this occasionally, as POCO3 reacts vigorously with water. And if you don't stir occasionally and allow the beaker to heat up, an explosion could occur, and you really do not want that. And so after you neutralize all the excess POCO3, we're going to slowly add sodium hydroxide to raise the pH to 7, which is a neutral pH. And we do this so our product will be nonpolar or non-charged and will be able to be extracted in a nonpolar solvent or a nonpolar liquid. So the next step would be to purify and isolate the product. So you would take the contents of that beaker, add it to an extraction funnel, and then you'll basically add the organic nonpolar solvent of methylene chloride. And so once you add both of these in, you're going to cap it here, you're going to invert it a few times, and then what you should see is a clear separation of phases. So this top phase here is the aqueous phase, and this bottom phase is the nonpolar organic solvent, because methylene chloride is more dense than water, so it will be on the bottom. And this is the fraction that you want to collect. So basically you are going to drain this into a flask and repeat that whole process again 2 to 3 times to ensure that we have all the product that was in the aqueous phase that we have now collected. Next, we are going to dry it, so we are going to use magnesium sulfate, and this is basically to make sure that there's no water present in what we collected. We're going to add magnesium sulfate, let it stir for a bit, and then we will filter that off. And then afterward, we will collect the filtrate, and then put it in an evaporation flask, and then place that in our rotovap. And what a rotovap does is basically distill off the organic solvent, so that all we are left with is our product, which, like I said before, should be a yellow oil. So how do we know what we collected is the structure that we want? We characterize or determine the structure of our collected product using NMR spectroscopy, proton and carbon-13 NMR spectroscopy. So these are a few spectra. I just want to point out here, these are not overlapping. These signals basically resemble this as well, but the main difference is these 2 peaks indicate the presence of these 2 hydroxy groups. And these peaks are not present in this product spectra that we have just synthesized. So that indicates a change in structure. And since the hydroxy groups are lost, we can assume that they have been replaced with chlorines. And to further this, I want to go into the next step, which this is step 4. So we are basically hydrolyzing this ester of the side chain. And these, trust me when I say that, these basically match that. The main difference being that this peak here indicates the presence of a carboxylic acid proton, which is shown here. So it indicates that we have successfully hydrolyzed the side chain into a carboxylic acid. And so, as you can see, there's a lot more that goes into a reaction arrow than one would think. We are currently on step 7. We have taken NMR samples of what we have done with this reaction, and we believe to have the product of step 7 in our sample. And so theoretically, we are just 2 steps away. So another reason why this research is important is that the current treatment for HIV is known as triple drug therapy. And patients undergoing this treatment have difficulties tolerating the toxicities of the drug, as well as these drugs are very costly. So by investigating alternative treatment routes, we have the potential to resolve these problems and perhaps come up with a treatment that is more beneficial for the patients. And so I'd like to say thank you all for listening. Thank you for my friends and family that came to watch me present. I would like to especially thank Dr. Kramer for being my honors advisor and being there for me every step of the way. I would also like to thank Dr. Stinsis and Dr. BRUNI for being part of my honors committee and giving me valuable feedback, and as well as the honors program for providing me with this opportunity to write an honors thesis. These past 17 months have probably been the most fulfilling and satisfying experience I've had at Millsaps. So with that, I would just like to say thank you all.
All right, do we have any questions for Christopher?
Christopher, I'm going to go way back to your backbone of the active sites. If I understood what you said correctly, the work that you're doing doesn't necessarily target those active sites, but it targets the backbone and the way that structure would inhibit the access to the active.
Correct, yes, sir. We are basically synthesizing an inhibitor to bind to an allosteric site of integrase, and that binding basically induces a 3D change in the protein shape, which will close off the active sites and thus not allow the integration of the viral DNA into the host genome to occur.
So this might not be a fair question, but nobody else asked, so I'm going to jump in. How do you choose which site in that complicated backbone is the one to attack so that the folding will actually inhibit that active site?
Yes, sir, that's a good question. So Dr. Kessel, who's the head over this collaborative project, he came and talked at Millsaps years ago, and he was looking for collaborators to basically synthesize these pharmacophores so he can test the efficacy of inhibition on his assays. And so his assays are targeting this specific site. So that's how we basically chose to do this.
Do we have any other questions?
Brian, curiosity, so I think you were on step 7, right? Is where you were at.
Yes, sir.
So just 2 questions from, when I was in pharmaceutical. How much product do you have at that point? Do you know what I mean? You have so many steps and we're trying to get something in. So about how much do you have there? How much do you hope to have it in? And second, are you testing the purity of it? Like I said, 97%, 98, 50, 50. Are you getting a percent yield? Are you cleaning that up to make it go high purity or just carry the impurities through?
Yes, sir. So to answer your first question, so these other steps progressively have lower and lower yields. That's why the optimization of these first few steps, so we have enough material to continue the synthesis, is so important. And we do, so the step that I was stuck on over the summer was this reaction here, and I purified it using a vacuum distillation, and that's when we got the purified product to that. And this product, we've run a few experiments with this. I'm not exactly sure how much we have, but it does show to be pretty impure, so we would need to purify this probably before moving on to the next steps.
Okay. How are you checking the purity, like TLC or?
TLC and NMR, so the presence of like a lot of peaks on top of each other would indicate a impurity.
And one last follow-up. So if it is impure when you go forward, you know, and then you don't clean it up, say prep out your product, how much does that inhibit the next synthesis? You know what I mean? You have unwanted stuff in there. How much of a problem is that?
I've not done a step with an impure reactant. We typically try to purify it. Me and Dr. Kramer have been thinking about whether we should just go ahead and continue this, as it may be easier to purify and isolate this product here compared to this product here. So we may go ahead and try to add on that T-butyl group shown here and try to purify it whenever that is added on.
Do we have any other questions? Okay, if there are no more questions, everyone please give another round of applause for Christopher. All right, and our next presenter of the morning is KAYLAH Scott with an honors in sociology, and her thesis is titled, "Did Natural Disasters Increase the Likelihood of Gentrification? Examining the City of New Orleans Post-Hurricane Katrina." Thank you.
All right, thank you, Hannah. Hi, everybody. Again, my name is KAYLAH Scott, and I conducted my research just looking into the relationship between natural disasters and gentrification, specifically Hurricane Katrina of 2005 and the city of New Orleans and a couple of its neighborhoods. So as we all may know, Hurricane Katrina made landfall. It began in the Gulf in 2005, and it made its way through states such as Louisiana, Mississippi, and Alabama. I will be focusing on its impact on the city of New Orleans for this thesis. However, it's been about 16 years since Hurricane Katrina made landfall, and it flooded about 80% of New Orleans and left almost 135,000 homes destroyed. So that caused a lot of people to have to evacuate. A lot of homes were lost, and New Orleans since then has not reached its pre-Katrina population. A lot of people, when they lost their homes, they decided to never come back. So there's been this sense of like displacement and pushing out where people never made their return. So that's what sort of struck my interest to pursue this as my research topic. So a few key focuses since the beginning of my research and towards the end have been whether or not New Orleans is a gentrifying city. And if it is, how much of this is a result of Hurricane Katrina 2005? So that's where you see where I ask if there's a relationship between natural disasters and gentrification. And if there is this relationship that exists, how do local residents feel about these changes that they see happening within their neighborhoods? Do they have positive or negative reactions or no feelings towards these changes at all? And if so, should sociologists have been included in rebuilding efforts from the jump? That's a key focus. So the methods that I've chosen for my research is a literature review. When you conduct a literature review, you read past research and works from other authors and researchers looking for some common themes within the research. And one thing that I have noticed while reading about the sociology of Hurricane Katrina, many researchers, one critique that they had is that when, city policymakers and different, architects and rebuilders, when they were trying to rebuild the city beginning in 2006, some people feel that sociologists and other social scientists were left out of the picture. They weren't there to consider the social dangers and risks and benefits of the rebuilding process. And that leads you to question, what if they were? What if someone were there to consider how residents would feel about the things that were done? So that's where, I picked up on that research, but first I had to conduct secondary data analyses and see, when you look at the numbers, if New Orleans and these neighborhoods are actually being gentrified. And after that, which the data did confirm that New Orleans is a gentrifying city, how do people feel about these things? So that's where I conducted qualitative in-depth interviews with residents that live in a couple of New Orleans neighborhoods. And after that, I took everything that I've learned with the data and with the interviews, and I took that and brought that to the actual sites. I conducted a field observation in each of the neighborhoods that I conducted my research on. So before we get into it, I first must define gentrification and what that is. So gentrification is this process of change that happens when there's an influx of certain resources within a neighborhood or area. And that can be through the influx of things such as people, homes, and businesses. And these new resources tend to, drive up the value and cost of these certain places. So you see things like mortgages and rents and property taxes and all of those things, those increase as well. And somewhere along that process, things become more unaffordable. People often lose their homes, can't afford rent or mortgage, and they have to move out. And there's this process of displacement and the pushing out of local and past residents. So when you try to figure out whether or not a place is being gentrified, there are often some key indicators that help you answer that question. One can be population decrease. As we've seen in New Orleans, like I said, we've never reached our pre-Katrina population. There tend to be less people living in these areas. Another thing is home ownership status. One thing that makes people vulnerable to gentrification is whether or not they own or rent their homes. There is a very significant relationship between home ownership and gentrification. You're more likely to be affected by gentrification if you rent your home and don't own it. Educational attainment. When it comes to gentrification and neighborhoods that are changing, you see that year by year, the residents of these communities are more educated and they have higher rates of educational attainment. That may mean that you have more people with college degrees or professional degrees of that sort. And there's also an increased cost of living. That means what that means. Rent goes up, mortgages go up, and it becomes unaffordable in the process. And finally, you have changes in racial demographics. With cities and neighborhoods that become gentrified, you often see that these are communities of color and ethnic neighborhoods, meaning that if you have a community that's, a black neighborhood and they're the majority, and you have, white residents, it often goes like this, meaning that the neighborhoods become less ethnic, and whiter. So that's what sort of sparked my research. I decided to conduct research in Mid-City and Treme. These are two very historic neighborhoods in New Orleans. and you may wonder why. But first, New Orleans is about 306 years old, but Mid-City, to say that it's one of the most historic neighborhoods in New Orleans, it's one of the youngest historic neighborhoods in New Orleans. It wasn't made habitable until 1913, and that's such little time compared to its other historic neighborhoods. There's been much change that took place in that city, so that's why I chose to conduct my research in Mid-City. And it's for Treme. Treme is the oldest black neighborhood in the United States of America. Some of you may not know this, but it was once a slave plantation, and once these slaves gained their freedom, they continued to make Treme their home, and that made it the first black neighborhood, one of the oldest black neighborhoods in the nation. So with that, I decided to continue with the secondary data analyses, just looking at those 5 indicators and seeing if numerically these neighborhoods are being gentrified. So as you see, this is Mid-City. Its population in 2000, before Hurricane Katrina, was about 20,000 people. And 16 years later, it has not reached that number, and it's still significantly smaller. Today, there are only 14,059 people that live in Mid-City. When it comes to rates of home ownership, you can see that Mid-City is a renter-occupied neighborhood with a whopping 74% of people that rent their homes rather than own them. And if you look, you can even see that there's been a decrease in home ownership in Mid-City. When it comes to the educational attainment, residents of Mid-City are more educated than they were before Hurricane Katrina. I have highlighted one of the most significant categories that I found. You have more people in Mid-City that have earned bachelor's degrees and completed college. When it comes to the cost of living, I found this pretty significant. As we know in the United States, in inflation, the cost of living will increase, but it's at the rate in which the cost of living increases. And I like to look to the United States right there. As you can see, the cost of living in Mid-City is increasing at a much faster rate than in the United States in general. So I found that pretty significant. And when it comes to the racial demographics in Mid-City, it is a community of color and it's very diverse. However, the black population, as I mentioned before, it's kind of decreasing while you see the neighborhood becoming whiter as the white population has increased from about 23% to about 40%. And that's for Mid-City. But when it comes to Treme, it's much smaller. There were only about 9,000 residents that lived there before Katrina. However, now, to this day, there are about 4,590 people that live in Treme. When it comes to rates of home ownership, Treme is also majority renter-occupied. Though there has been an increase in home ownership, there are still higher rates of people that rent their homes rather than own them. When it comes to the educational attainment of Treme, in 2000, if you see the people that didn't even complete 9th grade, that was 10.4%, but that dropped significantly. While the people who also earned bachelor's degrees, those rates have increased significantly. So you can see that the residents of Treme are more educated. And the cost of living. Before Katrina, as you can see, the average contract rent was $392, and now it's at about $714 compared to the United States, and how the cost of living changes, it increases at a much higher and faster rate. And this slide right here is the most stunning to me, because as I mentioned before Katrina, and since slavery was abolished in New Orleans, Mid-City was a very black neighborhood. As you can see, it had the residential population, 92.4% of the residents were black or African American. However, today, that has fallen to 56.3%. And you can see in the category of the white residents, only about 5% of the people that lived in Treme, they were white, but today that has risen to about 36%, so I found that very significant. So as you can see, as I conducted the secondary data analyses, the data and the statistics confirm that Treme and Mid-City are gentrifying neighborhoods for certain. However, with that information, there has been consequences in the housing market. There's a housing crisis in both these neighborhoods. And I wanted to see how the actual residents that live in both Treme and Mid-City feel about these changes and what they think. And that led me to conduct the in-depth interviews where I interview people that live in those neighborhoods. So the process of getting people to sit down with interviews for me, it was pretty hard. I remember in the beginning of it all, I told my advisor, Dr. Evans, that I wanted to interview about 25 to 30 people. And looking back, I can laugh at that because that was pretty ambitious. I actually ended up having about 15 participants that I talked to. However, the process of it all, I remember researching a few nonprofits and organizations that specialize in housing. And I called them up, email, and I said, hey, do you have people that you work with that might be willing to sit down for an interview? And they can just share their stories and thoughts on the neighborhood changes that they've witnessed and experienced. I also had a lot of help from my mom, as she helped me with community outreach and getting people to interview with me. And I also use social media apps like Nextdoor, where you can send out posts to people that live in these certain areas asking if they'd be interested in talking with me. So from my interviews, I had 15 participants total, and not only 12, but 12 of them allowed me to use everything that I've, seen in the data and use it in my written and published works. So of the 15 participants, 12 of them are Black and I had 3 white participants. And most of them were able to see that there was a relationship between Hurricane Katrina and the gentrification and neighborhood change that's been taking place. From their perspective, they saw that when you have, the floods that wiped out many properties, some people never came back, and there was, high rates of vacancy, and therefore that led outsiders and people who were no longer or never a member of that community. They came in and built houses, refurbished them, resold them, hiking up prices to where they were no longer affordable. I expected a lot more people to have pretty good feelings toward gentrification, but that's not what the data presented itself as, which is pretty shocking. Only one of my participants, which she was a Mid-City resident, she expressed positive attitudes toward gentrification completely, meaning that she was completely satisfied with the changes that she's seen in her neighborhood. However, there was a widespread disapproval of Airbnbs and short-term rental properties that exist, which is something that happens at very, very high numbers in both Mid-City and Treme, New Orleans. New Orleans sometimes is a tourist hotspot for Mardi Gras and other festivals and sporting events. So that's what they see in their neighborhoods. Outsiders coming in, spending the weekends, and just leaving at their own, at their own pleasing. So that was something that I found really interesting. And while I was talking to these people, they mentioned both a cultural and economic factor and effect that happens as a result of gentrification. Of course, the economic factor, things become less affordable, but they also say that the culture changes as you have new people coming in. It doesn't feel like home anymore, or it's not as communal as it should be. And I'll quickly let y'all see what some people had to say. These are some excerpts and quotes from the people that I interviewed. I have one woman named Rashida Fulton. She's 41 and lives in Treme with her 2 sons. She says, we have no savings. We live check to check and cannot afford to rebuild. So when a disaster happens, it's not an up for us. A lot of people think it's a big deal because the government give you a couple dollars here and there. But if you were already living check to check and you lose everything in your home, a $20,000 check is not going to get you everything in your home. Tim Wallace, a 27-year-old, he lives in Mid-City. While talking about gentrification, he said, and so gentrification is this sense of economic violence, and I would even say it's like modern-day colonization. It's like folks have discovered a neighborhood and they invest some money here, and that ends up driving up costs for the folks that have been living in that area. Whether that's property taxes, whether that's folks who have been long-time renters, driving up their rent. The folks who would have otherwise bought in this neighborhood, that have connections here. So there tends to be this displacing effect that happens because of gentrification. I interviewed a 47-year-old woman that lives in Treme. Her name is Christy Williams, and she made this comment on the people that gentrify neighborhoods. She says, and then they saw the opportunity there to live. They came in and saw vacant houses and said, ooh, let's move there since no one is here. Not much more mid-city than Treme because the cost of homes were a little bit more, but other areas like Central City, that's where the gentrification with living in the homes was a little more widespread because the homes weren't of great value. And there were more renters in the Central City than in Mid-City, which allowed the gentrification. Paige McGee of Mid-City, she's 22 years old, and she was my youngest participant that I talked to. And she said that everything is a tourist thing in New Orleans, and it's just like we can't be. People move here and build houses they couldn't even afford if they built them in the cities in which they came from. It's crazy. They're investing in a property, renting it out as an Airbnb, and making their money down here when they don't even live in the city, especially during Mardi Gras season. The different Super Bowls and football games, March Madness, and the festivals that happen in New Orleans. It's just a big business thing that's happening here, and a lot of people don't focus on the history of the city. So as you can see, most of my participants have this disapproval and negative feelings towards gentrification and the changes that come with it. And after that, I decided to conduct a field observation in these neighborhoods. Mainly the difference between the gentrification that happens in Treme and Mid-City is that in Treme, it's really focused in the housing of it all. You see a lot of vacant properties that were left there after Katrina, and people, they build houses and resell them. So here's a picture of some new houses that I saw in the area of Treme. And here are some of those vacant houses that I can predict that would soon leave. I also see some bike lanes. I'll just quickly breeze through these because I know I'm running out of time. People associate bike lanes with gentrification. And in Mid-City, it's more about the businesses that pop up and appear in these places. As you can see, these are local businesses that sort of exist in the areas that haven't been touched yet. However, there's been a recent arrival of things like Whole Foods and pizzerias in which people shop. And finally, the last picture that I have, I took a picture of this because I found it very captivating. You can see a regular plain house here that's probably been there for a while, yet in 2021, that condo that you see right there was recently built, and it doesn't even fit the classic New Orleanian architecture in which we see in the city, so it looks very much so out of place, being a physical indicator of gentrification. So that includes my research. Thank y'all for listening. I'd like to give a special thanks to Dr. Evans, my first reader and advisor. I'd also like to thank Dr. Belil, my second reader, and Dr. Sue Carey Truman for being my third reader. I'd like to thank Dr. Rice for being the director of the honors program and putting on the schedule for the weekend. And I'd also like to give a special thanks to Dr. Ming and Dr. Tordella Williams. They're no longer professors in the Millsaps community, but they helped me get my project off of the ground. All right, thanks.
All right, does anyone have any questions for KAYLAH?
What would you say was your biggest challenge in the interview process?
Okay, I'll quickly answer that. The biggest challenge was talking about the things in which we talked about. I know I had a few participants that had to pause sometimes and like recall and recollect on such a traumatic experience. So just being there, having to listen to that and like witness it in real time, it was pretty eye-opening. Because this is such a sensitive thing to talk about. So I would say that was the toughest part, getting people to actually like respond and say something while they, maybe held back tears or, very traumatic memories of their Hurricane Katrina experience. Yes, sorry. Yes. Yes, so I actually did glance over some things like that. And like you said, communities of color in ethnic neighborhoods are more likely to be vulnerable to things like gentrification. However, when it comes to natural disasters and how it affects the character of these neighborhoods, that is what I decided to focus on more, specifically how natural disasters could exacerbate rates of gentrification, although that gentrification was already happening. But definitely I did compare it to other cities, and as time progresses, it is a fact that communities of color, especially in major cities that maybe draw in tourists and other, visitors, they are gentrifying at significant rates, maybe not as fast as certain cities, but definitely communities of color and other ethnic neighborhoods, yes.
Awesome. I think that's all the time we have for questions, unfortunately. Y'all can find KAYLAH later if you have questions to ask her, but everyone please give her another round of applause for now. And our next presenter for this session is Rose Ward with an honors in chemistry. And the title of her thesis is Investigating Polycyclic Aromatic Hydrocarbons as Biosignatures in the Martian Subsurface using GCMS.
Thank you. Well, good morning, everyone. So behind every research project is questions that the research sets out to answer or help inform the answer to. So I'd like to start by introducing the research, the questions behind my research. So first, if there is evidence of past or current life on Mars, would it be detectable? Is there reason to believe signs of life have been able to survive Martian conditions? And if we can detect signs of life and believe they've survived, then what environments should the search for life on Mars focus on? Where should we look? Well, first, let's back up a little bit. Why Mars? We've all seen the fascination with extraterrestrial life and Martian beings, and what's the basis for that? Well, Mars does represent one of our best candidates for finding signs of extraterrestrial life for a few reasons. One, Mars is Earth's neighbor, so in terms of space, it is pretty accessible. But more importantly, there's evidence Mars had a similar climate to Earth in the past, a climate that could support and sustain life. A part of this climate includes having liquid water, which, as we know, is necessary to sustain life. So how do we detect these signs of extraterrestrial life, since obviously there's no big Martians walking around? Well, we look much smaller at molecular biosignatures, which are organic remains or compounds that can indicate past or present biotic activity, where biotic activity means it comes from a living organism. So these compounds are organic, so they're carbon-based. Life as we know it is carbon-based, which helps because we're using something we're familiar with and know how it works, so it helps when trying to identify this past or present life in other planets. It's like having a blueprint set out for us. So, I want to research biosignatures on Mars. How do I do that since I'm not on Mars and I don't have Martian samples? Well, we do this with simulation experiments, meaning I recreated Martian conditions and environments in the laboratory. To do this, I first selected 4 different minerals, shown in the figure to the top right. From left to right, these minerals are hematite, milky quartz, gypsum satin spar, and shale. All of these minerals have been identified on Mars. Next, I don't know exactly what biosignatures could be on Mars, so I had to select representative biosignatures. The ones I chose were 8 different polycyclic aromatic hydrocarbons, abbreviated PAHs. They're on the figure to the bottom right, and from left to right, they are naphthalene, 2-methylnaphthalene, biphenyl, fluorine, anthracene, phenanthrene, anthrone, and finally anthraquinone. So these selected PAHs were added into the minerals to simulate biosignatures in Martian minerals, and then the mineral PAHs mix was placed into different fluid environments. So these fluid environments consisted of a saline solution made from sodium chloride, an acidic solution made from hydrochloric acid, and then an iron solution from ferric oxide. Also, a sample was kept dry, so no fluid environments added. So to recap, for each mineral, there were 4 samples. 3 of them had fluids, 1 was kept dry, all of them contained the pause. After the samples were created, they were placed in a heating furnace for a cumulative 200 hours at 150 Celsius. After this heating time, the samples were prepared for analysis after being extracted. So, before I continue with analysis and talking about results, I want to give more background on why these experimental parameters, as this is a vital part of the research when doing simulation experiments. As I said, the chosen minerals have been observed on Mars, but the reason for even considering minerals as a host environment in the first place is partly due to the fact that previous Martian analog experiments have shown that properties of minerals can contribute to high preservation potential for biosignatures. As for the fluid environments, I said Mars has previously hosted water, but what could be in that water? Salt deposits have been observed on Mars that were formed when saltwater oceans evaporated and left the salt behind. There's evidence of past fluid conditions on Mars, and there are still current acidic conditions today, even though they are not fluid. However, there is a tricky thing with these acidic conditions, as they could be destructive to biosignatures. Finally, Mars is covered in iron minerals and ferric oxide, which is an oxidant of iron. This is what gives Mars its famous red color. But also, unfortunately, these iron minerals and oxidants could also be destructive to biosignatures. So PAWs were chosen as the representative biosignatures because, 1, they're organic compounds, so they are carbon-based, but more important, they are very stable and resistant to heat and pressure. And it will be clear why this is so important in a moment. Further, PAWs have been detected already in the interstellar medium, such as on asteroids and on meteorites. So we know PAWs are already in space. Now, here's an important distinction. Not all PAWs are biological in origin. So saying that we found PAWs in space does not mean that we've already found life in space. That would be very exciting, but it hasn't happened yet. As I said, PAWs are representative biosignatures, and analyzing these organic compounds and how they fare in Martian conditions is still very important when looking for signs of life. So next, why the high temperature? There's evidence for past hydrothermal systems on Mars, which are essentially hot springs. Other Martian analog experiments have found that these hydrothermal systems can confine and preserve biosignatures, which leads to the last thing, the subsurface. The surface of Mars is largely inhospitable to life in organic compounds. So to find potential biosignatures, our best bet is to look in the subsurface within minerals in environments that have previously hosted these hydrothermal systems. So now we can begin to talk about analysis and results. After samples were subject to the simulated environments, they were analyzed by gas chromatography mass spectrometry, abbreviated GC-MS or GC mass spec. The GC mass spec is an instrument highly effective in separating and analyzing volatile organic compounds, which is exactly what the PAWs are. Analysis with the GC is completed in 2 steps. The first one being chromatography, where the sample is injected into the GC and individual compounds in the mixture are separated. Then those separated individual compounds enter the mass spectrometer, where they are identified and analyzed by their mass fragments. The output of analysis is the chromatogram. On the figure to the right, you can see the chromatogram of all 8 PAWs that I used for my experiment. These PAWs were not subject to any experimental conditions. They were all added together in a mixture and then injected into the GC mass spec, and you can see how effectively it separated each of these compounds. So now we can discuss some results. This is a table summarizing the results. The columns across the top are ordered by the PAW compounds, and the rows are ordered by the sample environment. A blank cell indicates that the PAW compound was not detected for that sample by the GC mass spec. And DP stands for degradation product, where degradation product essentially means that a piece broke off of that parent compound. So when I say a DP was detected, I mean that piece was detected, not the full compound. So there are a few important things to note here. There are a lot of blank cells. This could partly be due to uncontrolled factors within the minerals, such as porosity, grain size, among other factors. and also the destructive conditions with the acid and the iron oxidants. However, this wouldn't fully explain why so many compounds weren't detected. Another thing to note is that shale had the highest number of compounds detected, where shale is the sample in the last few rows. This indicates a variation between the preservation potential of the minerals, with, for this experiment, shale having the highest preservation potential. Finally, even though there are a lot of blank cells, anthraquinone was detected in almost every single sample, which anthraquinone is the compound on the far right. So these results now left me with more questions. Why am I not detecting more compounds, and why is there so much anthraquinone? So to help answer these questions, I conducted a couple of supplementary experiments that yielded some very exciting results. First, the case of the missing pause could simply be because they evaporated off of the sample while the sample was in the oven. There was an indicated correlation between the number of times a sample would dry in the furnace and I would re-add the fluid environment and the amount of PA compounds detected by the GC mass spec. So, the fewer times the sample dried and was re-wetted, the more PA compounds were detected by the GC. This is likely due to the smaller compounds having a lower molecular weight and fewer intermolecular forces, meaning it would be easier for them to evaporate. In contrast, anthraquinone had the highest molecular weight out of my selected PA compounds, so it would be much harder for it to evaporate, which could also maybe help explain why anthraquinone was detected so much. However, there is another factor with anthraquinone. It was detected in a sample that it was not put in. This eventually led to my hypothesis that the other PA compounds were potentially oxidizing into anthraquinone. So if you look to the figure on the right, you will see the structure of anthraquinone. It has 3 rings with 2 oxygens attached. Now, all PA compounds are composed of these rings, though they can have different numbers of these rings. But if I had a 3-ring PA compound structure and it was oxidized, it would gain those oxygens and then become anthraquinone. So, to test this hypothesis, the mineral environment experiment was recreated with a small change. Anthraquinone was not added to the samples, and anthrone was not added to the samples. Anthrone was not added as it is known to readily oxidize into anthraquinone. It has a very similar structure. So as to not skew results, I also excluded it. Now, once again, as you can see, anthraquinone was still detected quite a lot. In fact, it was detected in half of the samples. So this gives evidence to my hypothesis that the recurrence of anthraquinone could be due to other PA compounds oxidizing into anthraquinone. So, finally, here is a summary of the main results from this research and how these results could help inform future Mars missions and the search for life on Mars. In short, there are variations between the preservation potential of minerals. Drying and re-wetting cycles need to be considered when searching for biosignatures, as compounds could be lost to the atmosphere. And it is important to consider oxidation products when searching for biosignatures and not only focus on the parent compound. Finally, there are a few suggestions for future research, namely considering the effects of pressure and further characterizing the mineral variations, as well as further exploring possible oxidation pathways. Right. So thank you.
All right, does anyone have any questions for Rose?
Can you repeat like the last sentence that you said? I'm sorry.
So I said, if we have protocols, we only detect a very limited number of things. Do you think that anthraquinone would be a good candidate for detection, since not only are you detecting it, but it could also possibly detect, as a degradation product from many other things that might have been out there?
I would say that it would be a good candidate for trying to find these organic compounds, but I would also argue that it appears in this research we saw a lot of compounds not get detected, potentially because they were evaporating off of the sample, and it appears that anthraquinone was big enough that it didn't evaporate, and also it had the contribution of being oxidized into. So I would say that we should, yes, look at anthraquinone, but also look at large organic compounds that would have an even better chance of being more stable and not evaporating off if we're looking at, possible past fluid environments. But I would say if we could only go collect a few samples and look for a couple things, I would not look for anything smaller than anthraquinone.
Any other questions?
Were you able to calculate the percent recovery of your compounds? Did you know that you had too much anthraquinone?
I didn't calculate a percent recovery specifically. So when I created the samples, I know exactly how much was put into the sample initially, so I could calculate approximately what that would be in part per million. And of course, I had standards that I could compare it to, but it wasn't like I specifically calculated how much was at the beginning and then how much was at the end. But observing the chromatograms, you can tell by the relative intensity between the standards and then the compounds compared to each other. So, if that answers your question.
Any other questions?
Seems like there might be a lot of good surface science to do in the different mineral structures to determine what kind of chemical activity or even adsorption and desorption happen in different environments. Does it tell you anything about what types of minerals you need to test, not just the target molecules that you're looking for?
It would, yes. I don't have as big of a strong as a background in geoscience specifically, but there is a lot of other people doing research on those mineral environments specifically because they are very important. And we saw even in my research where they weren't the focus and I wasn't characterizing a lot of the different properties, we were still seeing an effect the minerals had on the preservation. So trying to do these Martian simulation experiments, we can't simulate every single thing at once. We have to start breaking it down apart. So I started by looking at temperature, adding other things into it. So I would say the mineral variations are a very, very important part of this. And I think with further research, we could see that definitely a certain mineral is going to preserve better than another mineral will. We can already tell from this research, hematite is basically ferric oxide. That's not going to preserve biosignatures as well as shale did, which is a fairly inert mineral. But yeah, that is going to be a very important part, I think, in the future when looking for biosignatures on Mars.
Any other questions?
Yeah, just to follow up, that was what I assumed Dr. Dunn was going to that too, is that I'm presuming that you're saying that this would, since we are somewhat familiar with Mars surface and mapping, that this would direct where to send probes, right? And you indicated that some mineral environments have a much greater potential for preservation than others. Would that factor into where they would look at it?
Oh, yes, I'm sorry if I misunderstood that point, but no, that is, one of the motivating questions behind this is we need to know what environments that we should be looking in. So, of course, we have different ways that we can scan the surface of Mars and detect certain minerals that could be in different places, and I definitely think that we should be using that as kind of a guide to where to go on Mars and where to sample for potential biosignatures. So no, I do agree that should be a leading way in where to land probes on Mars, so to speak.
Any other questions? Well, if there aren't any more questions, everyone please give another round of applause for All right, that was our last presenter of this session, so we're going to have a 15-minute break, and we will start back at 12 p.m. for our last three presenters of the conference.
right, good afternoon. All right, good afternoon. Everybody ready? So I want to welcome you all back to the presentations after the break. My name is Faith Gilmore, and I will be moderating our third session. And it is my esteemed pleasure to moderate the last three for the day. And we have Brailey Vine, Anna Grace Box, and Nazem Rahat that will be doing their presentations. And so we have the last 5 minutes. I'll be sitting to your left. And if you just glance over, and I'll give you, if you go over like 15 minutes, I'll let you know. So we have first up Brailey Vine doing an honors in English. And her title is The Strange and the Familiar, Language and Home. Let's give her a round of applause.
Hi, my name is Brailey Vine, and my honors project is in English. Okay, so I sort of took the research project model and tweaked it a little bit to my personal interests. So I did do research into linguistics. That's what I wanted to do the project on. But I also did research into the craft of creative writing. I am a creative writer at heart, and so I wanted to produce creative content for this project. So I read books on how to produce successful creative essays, how to use literary devices to achieve a certain effect in the reader, and etc. And then I combined that and what I had learned in linguistics to create mini memoirs that examine linguistic concepts that I've observed in my own life. And I had the privilege of having Kelsey Burgesson as my third reader. She's a PhD student in linguistics at UT Austin, and she gave me great reading recommendations throughout the project. So first, I emailed her about topics I was interested in, and she sent me articles on them. But then when I started writing the essays, I would email them to her, she would read them, and she would respond with things like, hey, it seems like you're touching on this linguistic concept without really naming it, so here are some articles you may be interested in looking at to learn more and things like that. So working with her was a really great experience, and I'm really grateful that I had the opportunity. I'm indebted to both her and Dr. Pickard for putting me in touch with her, so thank you. For the first 2/3 of my presentation, I'm going to be reading to you in its entirety one of the essays that I wrote. So I'm hoping that hearing my work will help to justify why I chose to write creatively, i.e. I hope it's good and you like it. I didn't do a strictly academic honors project, so I want to demonstrate that I express myself and my ideas about the world best through creative nonfiction. So without further ado, I'm going to read to you an essay entitled Spanish Shaped. My mom often says I was miserable until I learned to read. I remember longing to not just look at books, but truly mine them for story. One of my earliest memories is of sitting at a low, round plastic table at the public library, discreetly watching over my picture book as a slightly older little girl with a messy part in her hair raked a finger across a tall page of Busy Town, directing it beneath the paths of the words. She seemed over-intent, and I wondered if she was pretending, as I often did. "Can you really read?" I asked her. I can't picture her lips in movement, can't recall her face at all. I know she said yeah, and I know I felt that sheer surrender to envy unique to childhood. What I really remember, though, is that moment of asking, of looking up from the shape of words at the uneven white part in her brunette hood, this hunched girl who could look at the same shapes and see candy, clocks, caterpillars, characters, colors. I did eventually learn to read and read all the time. For a while, however, I did not read very widely. So when I sat down in my third ever hour of high school in my first ever foreign language class, Senora Taylor handed me a sheet of paper that transported me from my cold desk all the way back to that warm plastic chair in the public library. I was face to face for the first time in a cloistered life with printed evidence of a language I did not speak, a language I had hardly heard outside of my young adventures with Dora the Explorer. Words were just shapes again. Even weirder, since Spanish uses the same alphabet as English with only minor additions, I felt like if I focused my eyes hard enough, I'd finally see story. Spanish words just looked like meaningless English ones, but then they didn't appear jumbled and random like key smashes. They looked tight, sleek, ordinary. More precisely, they looked like they would look ordinary to someone else, since they were certainly foreign to me. And orderly. They looked like they followed rules, which of course they did. And I spent the next 3 years learning many of those rules. 9th grade was 6 years ago now. That's at the time of writing this, not currently. And I'm considering declaring a Spanish major in college rather than just minoring in it, which I did end up doing. I love the language, adore learning words that have no exact English counterpart, such as sobremesa. Taken literally, this is a compound that says over table. And when I noticed that it was the word of the day on Spanish Dict once, I predicted tablecloth. But what it means is after dinner conversation. What can we extrapolate about culture from the fact that there is a word for those stories and jokes and concerns shared over streaked plates as unhurried hands pass sugar for coffee? Another of my favorites is rabiar, because it can mean to have rabies, to be furious, or to yearn for. I love it because it makes our anticlimactic English have rabies into an action verb. If a dog is foaming at the mouth, your Spanish suspicion is that he may be rabying. You can also raby because the airline lost your luggage, or raby for a kiss from your long-distance lover. I become unduly excited when I see no direct translation in its bolded type above a dictionary entry. And in case I've sparked curiosity, tablecloth is el mantel. I love speaking Spanish with natives. If they're older, they perk up hugely at the simplest sentences. An employee at my dad's restaurant once asked me if I could name the days of the week, and seemed so delighted when I could that I proceeded with the months of the year, and she taught me a song to go along with them. At my sister's graduation party last summer, I listened for 3 hours to my dad's friend's wife's stories about the milagros she's experienced as a result of her religious faith. On the other hand, though, if they are closer to my age, they often seem embarrassed or defensive, as if I may try to make fun of them for knowing both my native language and the one I am desperate to learn. This makes me want to cry, scream, roll on the floor. How anyone could disdain a polyglot for their abilities, I cannot imagine. ¿De dónde eres? I asked a friend of a friend upon meeting her. She raised both brows over defiant eyes and replied, Soy de Memphis, pero mi familia es de Mexico. All I could hear was, I'm from here, you bitch. I cowered. Of course, I wanted to say. Of course you belong. I just wondered why I understood your Spanish so well. Most of my teachers have been Mexican, so I recognize that accent best and speak with it as well. Interesting that she knew what I meant to ask. Where did you get your Spanish from? Mi familia. But it's crazy to me that she thought I asked it to condescend. How could I, the one tripping over basic conjugations in my second language, be seen as better than someone effortlessly thinking in two at once? My dad, reliably fascinated when his kids understand things he doesn't, will often push me to talk to people he knows or ask me to speak Spanish for just a dinner party of exclusively English speakers to hear. He once texted me a screenshot left on his restaurant's Facebook page written in a language I bet you can guess. What does this say? He asked me, with his signature ominous smiley face. So I understand a fragment of what bilingual folks must feel often. Like people want them to perform. Like they are an anomaly to be pointed to. And a finger in your face never feels like love, even if it's for being exceptional. But no one will ever disdain me for speaking Spanish, because my hair is blonde, my skin is white, and when I speak English, it is with an American accent. I cannot imagine what it feels like on the other side. I'm not yet comfortable describing myself as bilingual, because while I can carry on a conversation with most Spanish speakers I encounter, the more I learn how much there is to learn. And the learning is so messy. I think that the phrase pick up a language is accurate because you really must spy the crucial pieces for yourself and then push them together into something cohesive. When you're learning but aren't a little kid, people don't point to things and say their names. They speak. They throw sentences out to shatter on the ground like handfuls of parade candy, and whatever you pick up, you can keep. A new tongue does not mother you with carefully chosen lessons, it smothers you with everything at once. You and this language are like two adults meeting for the first time. To some extent, you start with the basics, but you meet as two wholes. And the obviousness of this looms heavy on both sides. You have all these crisp, sharpened concepts in your head, unlike a toddler. But you're less adept than a toddler in some ways, because often you don't even understand. Sorry. Speaking of toddlers, please indulge me by sitting through a few more paragraphs about my young self, and it will be over very soon. When I first learned to talk, my favorite word was a slur of two. What's that? I had a thick sticker book, and I would exhaust uncles, grandparents, family, friends, pointing to depictions of oranges, trees, chickens. What's that? What's that? What's that? My mom tells me that when I was 15 months old, she started referring to me as a sponge because on the way home from daycare one day, I pointed to a stop sign and said, "Mommy, look, an octagon." When she, impressed, posed an inquiry about the daycare's curriculum, she found out that there was none. But actually, Miss Sally supposedly said, I see her watching when I tell the older kids the names of the plastic shapes that go in this puzzle toy we have. That day when we got home, Mom drew various polygons for me and I named them all. She also drew a half moon, and I squeaked out, "Crescent." From the next room came my dad's voice, "The hell's a crescent?" That's probably the most impressive thing I've ever done. I did not, in fact, turn out to be a prodigy, and I am more than okay with that. The point is, my second language renders me a child again. I try to eavesdrop on snippets of Spanish conversation I hear in stores, and I can usually make no sense of them without the backbone of context. I lose track of storylines spoken in English, too focused on the subtitles I always have in Spanish. In classes, I look up from papers that contain both shapes and words to annoy professors with random questions that pop into my head. ¿Cómo se dice caterpillar in español? After college, I hope to move to Mexico. I want so badly to be a part of this world, to belong in Spanish the way I do in English. I raby for it. I hope that one day, no Spanish word I encounter is just a shape whose name I don't know, like crescents once were to my dad. As I tried to appear at 4, like a child who could read, I hope to one day pass deceptively as a Spanish-shaped person, a person whose Spanish has shaped their whole life. Okay, so for the second part of my presentation, I'd like to talk just a little bit about one of the linguistic ideas, concepts that I examined in my project. In one of my more academic essays, although even the academic essays I wrote were more philosophical than anything else. So the Sapir-Whorf hypothesis, sorry my graphic is blurry, my PowerPoint skills are subpar. So this is one of the most well-known hypotheses in linguistics. The idea is just that the languages that one grows up surrounded by have an effect, like a big effect, on the way you perceive the world. So I'm going to read just a couple more paragraphs that I wrote about this hypothesis in order to share the conclusions I came to. And if there are any little things in these couple paragraphs that don't make sense, it's just because they're taken out of context. Okay. The Sapir-Whorf hypothesis claims that language determines the way people perceive the world and think, rather than the other way around. Every linguist, from amateur to expert, must contend with this hypothesis. Yet I find that it's much more useful as a fluid idea than when we take a side, viewing it as either conclusive or discardable. I don't think we experience things through a linguistic lens. I believe we use our 5 senses to take in the world around us, unadulterated by the filters that make us insiders or outsiders when we speak. But I also believe that language has a rich life within our minds, and not just our interactions with other language users. So the turnaround between intake of an experience and output, i.e. framing for ourselves or others what something was like, is so quick that we easily disregard most things that cannot be put into words. That is, language doesn't determine how we experience what we experience, but its influence on how we remember that experience is heavy. We must learn language as toddlers, manipulating a sea of vocabulary with far greater aptitude than we can control our own feet, hence the label. So language is clearly acquired. Toddlers are certainly human, Yet what are the multitudes we contain if not knowledge funneled readily into words? The human spirit is vast, yes, but it is no coincidence that memory begins with our mastery of language. And what is identity made of if not memory? I argue that something very close to Sapir-Whorf is true. The human mind mistakes inexpressible for unimportant. We only notice the huge, jagged incongruities between experience and our ability to describe it when something important yet ineffable happens to us. But why? Why do we disregard the small, unsayable parts of our experiences from our own memory? Just because we can't describe something to someone else shouldn't mean we can't revisit it in our own mind, right? This is where the hypothesis would seem to hold some weight. Perhaps we literally are experiencing things through a lens our native language has created in our brains. But I don't think so. I think we are such communal creatures, relying so heavily on connection to other humans for validation of our own humanness, that we discard pieces of memory that resist language so long as they are largely unnecessary, because they isolate us from others. We value only those thoughts, feelings, and experiences that we can turn into language because we value only what can be shared. And finally, I owe thanks to all of the people listed here, but I'd especially like to thank my third reader, Kelsey Bergson, whose input was so valuable at every point in the project, my second reader, Dr. McMaster, whose edits and suggestions are always so thoughtful and genuine, and my first reader, Dr. Pickard, who has been my advisor and therefore putting up with my antics since freshman year. I owe that man a debt of time and effort that I will never be able to repay, and because he does what he does out of a true love of learning and mentorship, I know he'll never expect me to, but I wish I could. Thank you.
All right, do we have any questions for Brailey? Yes.
What made you want to take a more creative approach with your honors?
I don't really like research. And I love creative writing, and that's literally the only reason.
Any more questions for Brailey? All right, well, let's give Brailey a round of applause. All right, next up we have Anna Grace Box conducting her thesis in biology titled An Analysis of the Effects of Coastal Restoration and Feral Hogs on Malaclamese Terrapin Nesting Ecology. Let's give her a round of applause.
Hi, just a second, I'm trying to find my, here we go. Hi, so my name is Anna Grace, and my honors thesis was an analysis of the effects of coastal restoration and feral hogs on Malaclamese terrapin nesting ecology. So first I'd like to tell you a little bit about the species that I worked with. The diamondback terrapins, or Malaclamese terrapin, they're the only brackish water turtle. And if you're unfamiliar with that term, brackish water is just the salinity level between freshwater and saltwater. They nest on sandy soils on beaches, typically, above the high tide line. Now, their global conservation status, as listed by the International Union for the Conservation of Nature's Red List, is vulnerable. Now, what this means is that they are susceptible to global extinction. On the state level, they are listed as a Tier 2 species in Mississippi, and this is by the Mississippi State Wildlife Action Plan. What this means is that they are in need of timely conservation action. However, this has not been updated since 2015, and it's possible that they require a new conservation status, which I will touch on later. Now, two previous studies that I will discuss later on are Mann 1995, which Dr. Thomas Mann performed a series of nesting surveys along the Mississippi coast. And Dr. Coleman, 2012 to 2014, also performed a series of surveys throughout the summers for those years along the Mississippi coast as well. The goals of my study were to examine the impact of restoration and the subsequent changes to the nesting beaches on terrapin nesting, as well as to update the knowledge of terrapin population in South Mississippi. The Mississippi State Wildlife Action Plan that I previously mentioned does indicate that there is a high need for the conduction of population estimates in South Mississippi. So I think this is a valuable contribution to that field. My study area is Point Clear Island in Hancock County. This includes the northeastern portion of the Hancock County Marsh Preserve. My study area included the shoreline, which was a 2.44 kilometer stretch of beach. It is bounded to the north by the Bayou salt marshes, to the south by the Mississippi Sound, to the west is Bayou Bolin, and to the northeast is Waveland, Mississippi. Now, I will be mentioning several sections 1 through 5. Those are just these listed here of the study area. So here are some aerial images of Point Clear. As you can see in that first image, that is July 2005. This was a month before Hurricane Katrina. As you can see, especially in this area, the marshes were very healthy. There's a lot of vegetation. The beaches are fairly small in width. On August 29th of 2005, that was the day that Hurricane Katrina hit. As you can see, there is significantly less vegetation. The width of the beaches is thicker. And in September of 2010, you can see that same area, there's been a lot of erosion of that coastline, as well as on other areas. Now, 2010 is the year that restoration took place in the area. And this restoration is the Bayou Marsh Restoration Project. Now, it was launched in 2010 by the Mississippi Coastal Improvements Program, and it aimed to restore these marshes along the shoreline of Point Clear. They did this through a number of steps. First, they built up the land with dredged material. Then they planted some marsh vegetation, which included Spartina alterniflora, Spartina patens, and Juncus romerianus, which include some of these marsh grasses in the image behind me. They also placed geotubes along the shoreline, and those are those whitish gray structures that you see on either side of the screen. Now, the purpose of these are to protect from wave damage and storm damage. Now, after reassessment in 2015, they decided that they required more protection against storm damage and wave damage, and they installed a 650-foot long rocky breakwater and a 1,925-foot long living shoreline. And the purpose of these was just to provide extra protection. So here's some more aerial photos. This black and white image is in 1989. It was 4 years prior to Dr. Thomas Mann's study, so this was kind of like the baseline. In 2010, the next image, that was the year that restoration began. As you can see, there is evidence of the coastal erosion and the marsh erosion. And that image there is the restoration site. Now, in 2014, this was the last year that Dr. Coleman performed his surveys, so phase 1 of the restoration had taken place at this point. Finally, this was the most recent aerial photo that I could find. It was in 2019, 2 years before my study. All of the restoration had taken place, and as you can see, there is declining beach over time. This is one of those sections of the Bayou Marsh Restoration Project. Just to kind of let you know where we are, there's the geotubes, there's some of the planted vegetation. So as you can see, these are now mudflats and marsh. The beaches there no longer exist that terrapins once used for nesting. This results in a decline in beach of 0.631 kilometers. So this is likely to have caused a shift in the site selection of terrapin nests. So to investigate this change, I performed the following methods. I performed a zero nest survey on the 15th of May, and the purpose of this was to survey for potential nest sites, as well as to remove any nesting material from the previous season to prevent any false counts. I then performed weekly surveys. These ended up being 8 walking surveys between the 24th of May and the 30th of July. During these surveys, we searched for eggshells. As you can see in that top image, that's typically what they looked like. They were broken. And we identified a nest as having both eggshells and a nest hole. And the reason for that is the protocol that Dr. Coleman used in his survey, and I wanted these studies to be as comparable as possible. I then used my handheld GPS to record the location of the nest and to record other data such as predation around the nest and vegetation. Afterwards, I threw the eggshells into the marsh behind me to prevent double counting the next week. So here are the nests for my study. They are those very small red dots on the screen. So as you can see, the highest nesting density was in what I've called section 4, and this is where Dr. Mann in 1994 found the highest density. He found about 35 nests there. So this remains an important nesting beach in 2021. However, the next highest density that Dr. Mann found was where the restoration took place. He found about 13 nests there. And as I've stated, this no longer exists as a potential nesting site for terrapins. So there likely has been a shift. The next highest density was section 2, followed by section 3. Only 1 nest was found in section 1. This is because either the female terrapins are selecting against this beach. This beach was covered in sea glass and other debris, or the debris is covering the nests and protecting them from predation. So the only nests I found were depredated nests, as do most studies looking for terrapin nests, because it's just very difficult to find intact nests. And I did not find any nests in section 5. So here's some of my data. My mean clutch size, which clutch size is the number of eggs per nest. I found 5.9 plus or minus 3.28 eggshells per nest. Dr. Mann in 1994 found 8.25 eggshells per nest on average, and he found a range of 4 to 15. Now this is slightly less. This could likely be due to the high amount of hog predation I found. So 79% of nests I was able to assign predation, and all of these were feral hog predation, as far as I could tell. If there has been an increase in the hog population and an increase in hog predation, this can cause the clutch size estimate to be decreased because hogs tend to scatter eggshells and just completely obliterate the nests. So I found a total of 110 nests with 653 eggshells. Dr. Mann in 1994 found 98 nests, which at first glance seems pretty comparable, but he only performed 2 surveys, whereas I performed 8. So if you look at it that way, I found less than he found. And Dr. Coleman in the 2010s found 256 nests his first year, 441 the next year, and 377 the next year. So that is 2 to 4 times greater than what I found. So that was when we started to realize that we think that the restoration and the hogs have definitely played a role. It is likely that the decrease in the nesting beach and the shift in the nesting density has allowed it to the hogs to more easily find these nests and prey upon them. And we also kind of looked at Dr. Coleman's data. Here is on the y-axis the number of nests compared to the dates. So we looked at the peaks. The first year of Dr. Coleman, he didn't find a peak, but it was sort of headed for one in August. In 2013, there was a peak in late June and potentially in September. In 2014, he found a peak in late May and mid-July. And this very bottom one, which as you can see, significantly less nests, early June and early July. So we did not find that there was much correlation between the peak times over the years. However, it does appear that they are laying 2 clutches per year. As you see, most of these have 2 peaks. So as I've said before, I recorded the vegetation around each nest that I found. 77% of nests were near Spartina, which is cordgrass, and it looks like this. Other vegetation that we found was American beachgrass, seaside goldenrod, dollarweed, marsh elder, and brown-top millet. Now, the reason that this is important is that vegetation can affect nest site selection. So it can shade and cool nests, it can block avian predation, and it can also be a determinant that a nest is going to be above high tide line. And as I've said before, terrapins nest on sandy soils above high tide line. This is important because if anyone goes to do surveys later on in the area, these can potentially be used as nest site indicators. Now on to feral hogs. So as I've said, there has been very high predation that I found, and this can potentially cause a decrease in terrapin population or potentially extirpation, which is localized extinction. This can also cause an increase in the hog population because they're more successful in their hunting. Now, Dr. Mann in 1995 indicated that hogs are important nest predators in the Heron Bay marshes, which is part of the marsh system that Point Clear is in. Dr. Coleman, when I spoke with him, indicated that there was moderate hog presence, and in 2021, there was extreme, very high hog presence. The beaches were covered in tracks and these pits that you can see in this image. This indicates that there's probably been an increase in the hog population since 1994. This is important because these pits, these rooting pits that they make, can decrease the plant cover by up to 80%, and as I've said, vegetation is important for terrapin nesting. This can destabilize the beach habitat and potentially make these beaches unfit for nesting. So hogs have been a nuisance species in Mississippi since the 1980s. This is because they reproduce very rapidly and they have an adaptable diet. They are opportunistic omnivores, which basically means they will eat anything they can find. Their high fecundity makes it difficult to manage populations because they're reproducing so rapidly. You have to have a program that is going to decrease the population even more so. However, there has been success in small areas and islands, such as Santiago Island and Santa Cruz Island in the Galapagos. I think that bodes well for Point Clear Island, as it is both a small area and an island. However, this does require a bit of an investment because consistent efforts are needed afterwards to keep that population low. So future studies, I believe that there needs to be more observations on both the terrapin populations and the hog populations to investigate this relationship between the possible increase in hog population, decrease in terrapin population, if that's what's going on. I think we need to determine the conservation needs of diamondback terrapins and potentially upgrade them to a Tier 1 in Mississippi, which indicates immediate conservation action. As I've stated, this was last updated in 2015, which was near the end of Dr. Coleman's study, and as we've seen, there's been a lot of changes since then in terrapin nesting. Ultimately, I think that the conservation of terrapins in this area is going to rely on the implementation of a hog population control program. And that is all. Thank you.
All right, do we have any questions for Anna Grace? All right, let's give her a round of applause. All right, next up we have Nazam Rahat with her honors in biochemistry titled Substrate Specificity of Putative E. coli Antiporter YHJX. Let's give her a round of applause.
Okay, so if you've been here yesterday, you should be excited to hear the final saga in the protein expression story. I'm happy to be at your service and tell that little story for you. So my presentation today is on the substrates for transport of the E. coli membrane protein YHJX. The reason why I'm studying this specific protein is because understanding its function is important to understanding how E. coli has adapted different mechanisms to combat different levels of acid stress in different conditions. There are already 4 known systems in which E. coli uses to combat different levels of acid stress, and they all follow a very similar template mechanism. And we're going to look at AR2 to be our little example. So as you can see, glutamate, that's going to be the substrate molecule, and it is entered into the cell by a membrane protein. It then undergoes a decarboxylase reaction, and in this reaction, it forms GABA. GABA is then removed from the cell by the same membrane protein that brought glutamate in. This specific type of membrane protein is called an antiporter because it exchanges substrates across the membrane in opposite directions. So I am interested in looking at the oxalate-dependent system of acid response. It's a little different from the other ones because it requires an additional step in order to achieve the same basic result. So this is the proposed mechanism of how this works. Oxalate is brought into the cell by an antiporter protein, and before it can be decarboxylated, it has to be activated to form oxalyl-CoA. So it reacts with a molecule of formal-CoA to form oxalyl-CoA. Then oxalyl-CoA is, let me see if I can show you, never mind. Oxalyl-CoA is then decarboxylated to regenerate formal-CoA. It's important to note that in the byproduct of the first reaction, the transferase reaction, you're forming formate. Formate is actually the molecule that is exchanged across the membrane with oxalate. So we know the enzymes for this mechanism that conduct the transferase reaction and they catalyze the decarboxylase reaction. You can see them denoted by YFDW and YFDU. The structures of these enzymes and the kinetic parameters of these enzymes have been published. However, what's different about this system is that we have not yet identified what the antiporter protein is. Now, I'm interested because YHJX has been selected or hypothesized to be a candidate as this antiporter protein. The reason why is that it is homologous to another protein known as OXAL-T. OXAL-T does conduct this function, meaning that it exchanges oxalate and formate across the cell membrane in the bacterium Oxalobacter formigenes. Furthermore, we think YHJX could be this because you can see by this graph, it's called a hydropathy plot. All you really need to know is that this matches what a membrane transport protein should look like based off its relative polarities of its amino acid sequence. So now I've identified a candidate, a protein of interest to study, and the goal is to express, purify this protein, and use it in assays to determine if it does indeed exchange oxalate and formate. But before I can express that, I have to clone my gene of YHJX from the E. coli genomic DNA into an expressionable plasmid. So we selected PNH-TRXT. It's just like our vector of interest, and linearized it by restriction digest. This gel just shows that it's of the proper appropriate sizes. And so the 2 bands at the top are the plasmid. The 3 lower bands are PCR amplified products of my gene, YHJX. So now I insert the gene into the vector plasmid. Here just confirms that cloning was successful. Did a double digest, meaning I cut our construct at different fragment lengths, got our expected fragment lengths as visualized in that gel. So now I have my construct, and I transform an expressible strain of E. coli, known as BL21 cells, with my vector. And the next step is to start expression cultures. We did that, and you induce it with a molecule known as IPTG during a specific phase of the cycle. And IPTG just turns on protein expression, basically starts overexpressing your desired protein. Now that we've expressed these proteins or started expression cultures, harvest the cells, and can analyze our samples on an SDS page gel. So that's what you see here. SDS page separates proteins based off their sizes. So we expect to see a thick band of, just dark band of protein at our expected protein size. So if you look at the gel on the left, where I have the red boxes, that's where you want to see YHJX. That's the size of it, around 54 kilodaltons. Obviously you don't. So thinking that maybe expression levels were just really low, you can concentrate your protein, your samples through purification, and maybe see a more intense band. So if you look at the band on the right, at the same level, right where the red box is, now you should see a dark band. Again, you do not. So what went wrong? There are a few different things that can affect protein translation, one of which are mutations and the nucleotide sequence. So a nucleotide sequence of DNA is read in groups of 3. If there's an insertion or a deletion, it can change the frame, and that can mess up your translation of your desired protein. So we sent off our data, our construct, to be sequenced, and looked back and did an alignment of our construct versus what the sequence should be, and there was no frame shift mutations, meaning that there were no insertions or deletions, and everything looked all right. However, there are other types of mutations that can still affect protein translation, one of which being a nonsense mutation. It causes early termination of your amino acid sequence, meaning your protein's not at the right size. So that could explain why we didn't see a band where we were expecting to see a band on our SDS gel. So we looked at the amino acid alignment of just the sequence itself, and we did find 4 mutations. However, these mutations did not cause a nonsense mutation, meaning it did not cause early termination of our sequence. Just looking at the possible effects of what they could have done to the protein, here you have the same hydropathy plot from earlier. In the blue is the known sequence of YHJX, and the orange is our sequence, like construct, and you can tell there's very minimal differences in the 2 sequences, thinking that these mutations were not the reason why the protein did not express. So now the question is, why didn't the protein express? Can we express a protein? So we obtained a commercially available kit that we used in order to make a control protein from Bio-Rad Technologies. We tried to express the protein dihydrofolate reductase, and looking at their step-by-step procedure, their sterile materials, we were able to express that protein, which is good. It means we know what we're doing and we can do this. So this was at its expected weight of around 47 kilodaltons. So we also tried to express the protein of interest YHJX at the same time as this one, under the same conditions, now that we have a control. And unfortunately, you do not see the band of YHJX. So now other researchers have had success using a different medium. We've been using a standard medium known as LB to start our expression cultures. And we started using this medium called terrific broth. People have had more success with this, leads to higher density cell cultures, more nutrient rich. Used that in addition to, again, step-by-step protocol from the Bio-Rad kit, and once again, did not see the band where we wanted to. So you do kind of see a dark band here. It's a little lower than the expected weight, meaning it's not our protein. Also, that same band is in multiple other lanes. And those lanes are of different proteins of different sizes, meaning this is a non-specific protein, not our protein of interest. So we have been able to confirm a few different things. We know that the coverage due to our cloned vector is good. There's no mutations causing mistranslation of the protein, and we know that we are capable of expressing protein here at Millsaps. There are a few different things that we could do in the future to make sure and optimize our expression protocol. We could try doing it at a lower temperature. Other people have had success with that. Beyond expression, though, there could be some issues with our plasmid that are causing this to mess up. We could fully sequence our entire plasmid to ensure that the promoter and different parts of the translation, like machinery, and their sequence is working, and there's no mutations within that itself. We could also just restart cloning with another plasmid, change our backbone, because we've had low success with the PNHTRXT vector, but we were able to express the dihydrofolate reductase that had a different plasmid background. So lastly, I just want to thank the Millsaps Honors Program, Dr. Rice for organizing this, Dr. Kramer, Dr. BRUNI for serving on my committee, and then my little team, Dr. Toyota, Sam, and Connor. Thank you all for everything, because this was just so much fun to do. That's all.
Thank you. All right, do we have any questions?
The obvious question is, why wouldn't you have started with anything other than a product called terrific broth? That's all. My question is, the last set of slides that you showed, within your little vents where you said there's a dark path, a dark spot, that's not your protein.
Yes, so if you look at this one right here, you kind of see like the dark band where I circled it. But if you look at the other lanes, like lane 3, lane 7, and lane 9, those are all different proteins that we were also trying to express at the same time. And you see that band consistently as being like darker, so you would think it's our like overexpressed protein, but all those different all the different lanes are targeting for different proteins of different sizes. So this is showing that this is like a protein that is found, and it's a different protein other than all of these that is just being expressed at high levels.
We should study that protein.
That's what we're doing wrong. Sorry, guys. I'll come back next year and do it with this one. Saying a control with the same plasmid, the PNH, no, we haven't. We have all these, the multiple proteins here, GAD-C, YFD, W, YFDU, YJX, have all like failed expression, and they're all in the same plasmid. So the one that we use, DHFR, has a completely different backbone with a different antibiotic resistance, but it does have the same promoter system. But it is important that it's a different type of plasmid, though.
We have any other questions? Okay, well, let's give her a round of applause. So this concludes our third session of the Honors Conference for the day. So please let's give all of our presenters another round of applause. So in about 15 minutes, we will be conducting our award ceremony, so please stick around. And if that is it, okay, well, we're on break right now.
Okay, if I could have your attention, please. If I could have your attention. Thank you so much for waiting so patiently as we deliberated over these wonderful theses. So I'm Michael Pickard. I'm a professor of English and creative writing here at Millsaps and president of our chapter of Phi Beta Kappa. I just want to say a few words about Phi Beta Kappa so that you understand what this group is, and then I'll get to the good part. So Phi Beta Kappa is the oldest and most distinguished academic honor society in America, founded in 1776 at the College of William and Mary in Virginia. This society counts 17 U.S. presidents, 42 Supreme Court justices, and more than 150 Nobel Prize laureates among its members. In 1989, Millsaps was granted the first chapter of Phi Beta Kappa in the state of Mississippi. And every year, our chapter selects new inductees from among the class of graduating seniors. So we'll be announcing those new inductees for 2022 who meet the high standards for academics and character set forth by our chapter at TAP Day on Thursday, March the 3rd. So we also award prizes for the best theses and presentations delivered at the college's annual Honors Research Conference. So we begin with a selection based on the individual reports of the honors committees, and then we go from there. And this year, for the first time in our history, we've invited alumni of our chapter from across the country to join our faculty and staff members in assessing your theses and presentations. Before I announce the winners, I want to offer congratulations on behalf of all my colleagues in the Mississippi Alpha chapter of Phi Beta Kappa to everyone who presented research at this conference. In pursuing excellence of thought through so many diverse disciplinary fields in search of new and useful knowledge, you've made us proud, and we can't wait to see what wonderful things you will go on to do from here. So this year, the Mississippi Alpha chapter of Phi Beta Kappa recognizes Anna Grace Box for an analysis of the effects of coastal restoration and feral hogs, and I won't read the technical name, nesting ecology, for best thesis in the Division of Sciences. And we acknowledge and recognize Riley Houston, Civil War Nurses and the Cult of True Womanhood, for best thesis in the Division of Arts and Humanities. And finally, we recognize and honor Hannah O'Brien, Exploring Species-Level Diversity, again, names that I won't try to pronounce, in the mountains of Arizona, for the best honors thesis presentation. Please join me in congratulating these recipients and also all the presenters at this year's honors conference. So I'll need to huddle with the winners for just a few minutes after the presentation, but before we go, I also think that we should give a round of applause to Alex Rice, who has been the director of the honors program for this year, for putting everything together today. Alex, thank you so much. And I think that officially concludes our festivities. Have a great rest of your weekend, everyone, and thanks again for coming.